Hi everyone. Oh my god, just testing a mic. Okay, gonna revise biology today, so just gonna start with this very quickly. All right, Sophie. All right, Lucas, Adam, Yoshi, Sana. Who else is in here? The Egg Master's here. Okay, cool. Right, really quickly, just for those of you that have the nervous system coming up. Um, Very quick recap on the nervous system. So, what do we call the change in the environment? Anybody know what that is? This person stuck their finger on a pin. So what do we call the change in the environment? Thank you guys. Stimuli, awesome. Nice one. And then what do we call the cells that would be in the skin? Higher. Yep, well done, Eggmaster. That's the stimulus. What about the cells in the skin? Yeah, then we got the receptors. So these would be the receptors. And then we're going to have an impulse. It's going to travel down. What do we call the first nerve that the impulse travels down? So after the receptor, what nerve takes the message from the skin to the brain or spinal cord? Sensory neuron. Nice one, Ruby. Nice one, Lucas. Yep, everybody sensory. Awesome. And then you've got a gap in between the sensory and the relay. Anyone know what that's called? The gap between the sensory and the relay? Or the gap between any two neurons? Synapse. Awesome. You guys are on it. Awesome. Nice one. Um, so then we've got the relay neuron here labeled Q for some reason. Yep. And then another synapse. And then what neuron takes the impulse to the muscles? Yeah, I'll be going over meiosis. Yeah, I'll be definitely doing meiosis. I'll do whatever you guys need. So yeah, yeah, motor neuron, Lucas. Well done. And then the muscle. What's the general term for the thing at the end of the motor neuron, which can be a gland or a muscle? What's the general term? This is a striated muscle. The effector. Awesome. Nice one. So that you've got the effector there. Awesome. So you guys know your nervous system perfectly. I've got a couple of other diagrams here, so just give me 1 second. There's a couple. So watch out. This might confuse a few of you. Uh, Look at this diagram. This is one from an exam. It says stretch receptor there. So you have different types of receptors. That's detecting you stretching your knee. But look, on this one, he says, moving it all over the place. All right, I'm going to stop moving it around. I'm getting a bit like excited there. Okay, there I've got C. What is C labeling? So some people might put relay neuron. That would be wrong in this case. What is C pointing to? Mitosis was in paper one, but they could ask you a sort of comparative question where they mainly focus on meiosis, but mitosis is in the question. So I will run through a kind of versus mitosis and meiosis. Lucas, it probably won't come up, but I will do a quick reminder of mitosis just in case. The relay neuron is Q on this diagram, but C here, if they asked you to label this, that's the spinal cord. Yes, Gabriella, that's the spinal cord. So what you've got to watch out for is that the CNS, the central nervous system, contains the spinal cord and the brain, and relay neurons run through it. But you've got to be careful when talking about that. That there is labeling the spinal cord for C, not the relay neuron. The relay neuron runs through the white matter inside your spinal cord, which is surrounded by grey matter. You don't need to know about the grey and white matter, but yeah, just watch out for a little slip up like that. And then also some of you, depending on which exam board you do, so let me just move all of these over there. They're getting all a bit messy. I'm going to delete all of these actually. Can I delete all of this? Yeah, I can. Oh, I don't want to try and rub that out. That's just too long. So I've also got a diagram here of a neuron. Okay, what type of neuron is this? Looking at the information, um, so Miriam asks, when is it the central nervous system or when is it a neuron? So the central nervous system is your brain and spine, and the neurons that run inside it are the relay neurons. So if the diagram is asking you to label the relay neuron, you'll see it will be pointing to a line that's going through the brain, or normally the spine. In 99% of the diagrams, it's the spine. So that will be the relay neuron. When you're answering a 6 marker about a nervous response and you're just going through from receptor to the sensory neuron to the synapse, you normally can then say, then it's passed to the relay neuron, or you can say passed to the central nervous system, known as the coordinator also. So you can generally put that, but if they're asking you to label it, you clearly got to be clear that the brain and spine are different, or they're the central nervous system, but they're different than the relay neuron. Yeah. So this, what type of neuron was this? Did anyone spot what neuron this was that I've circled here? This is the sensory neuron. Nice one, Adam. And you'll be able to tell that because it's connected to the skin here. And also this thing, which we call the nucleus or cell body, is in the middle of the neuron. Whereas with a motor neuron, it's right at the end. Okay. Let's move. You guys are cool with all of that. Right. If you're doing AQA combined or AQA triple, if you're a biologist AQA or combined, and actually some of the other ones as well, you may be doing the reaction time test, the ruler drop test. So it's very probable that that could come up. Yeah, this won't come up for you, Adam, but we're only going to be on this for a minute. So, yeah, the cell body, broski, you probably don't need to know the cell body. The cell body is this thing I've circled here with A. That's the nucleus, by the way. A is pointing to the nucleus, but the nucleus is just located in the middle of a sensory neuron, whereas a motor neuron, it's right at the end. I don't think that'll be on your syllabus. Otherwise, you probably know that. So, we call this bit the axon, but cell membrane of the axon. If they're labeling this, the black line on the edge, it's the cell membrane. Oh, I'm going there too much. So yeah, you just got to watch out. That doesn't normally come up at all. Right, I've got a set of results here. Ruler test comes up a lot. I've got a set of results here with the ruler drop test. It depends whether you're doing AQA or EdXL, so it really depends which exam board you're doing. So yeah, I'm going to go through species. That's all coming up. Right, guys. First, student D. This is a ruler drop test. Which student here has got an odd? Which is the odd result? Which one of these is an odd result? For D, for student D, if you read across the 16-year-old with their reaction time, which one? Test 3, Broski's on it. He's been drinking his coffee tonight, so, or, yeah, 0.99. Yeah, absolutely. Now, what should the students do when they want to calculate the mean for student D? How would they do that? You're probably going to get tested on that. What should student do when calculating the mean? Good. Absolutely, you guys are on it. Well done, Lucas. Well done, Broski. Yeah, remove the mean and don't include it. So you divide by 3, add up the 3, divide by 3. So you just totally remove the odd result or anomalous result. Well done. And you may get one where they haven't done that and you have to spot they haven't done that. Like they've calculated a mean and you have to spot, hold on, their mean isn't correct because they didn't remove the odd result. So watch out for that. Also, a couple of things with the ruler drop test. Just be aware, if you're doing the ruler drop test, thumb and finger have to be the same distance apart. The ruler should be at the 0 mark. Very important, the ruler's at the 0 mark and that type of thing. In fact, I've got a 6 marker here on the ruler drop test. So this is the last thing on the nervous system before we jump off for those people that are like, hey, why are you doing nervous system? So describe a method to investigate the effect of caffeine on reaction time. So obviously caffeine supposedly, got my coffee here. Supposedly increases or decreases, decreases your reaction time, makes you quicker. Obviously, if the time goes down, you're faster. So yeah, caffeine should decrease your reaction time. Place arm on a desk, Claire. Absolutely. Yeah, 8 p.m. Asking for trouble, aren't I? Yeah, so place your arm on the desk, thumb and finger the same distance apart. Drop the ruler. Make sure it's random when you drop the ruler. You want to drop it randomly. Your partner. Make sure it's at the 0 mark. 3 times. Take an average. The shorter the distance, the quicker the reaction time. You can use, they might give you a conversion chart. You can convert the distance into times. So watch out for that. There might be some mathematical conversion chart, like each centimeter is 0.5 seconds or something. Yep. Good. Then, what else? You got issues with, there's loads of variables you can't control in this experiment, like, or it's difficult. What did they have for breakfast? Have they had caffeine earlier in the day? And that type of thing. So, and if you're going to do an experiment on boys, don't start making a generalized statement. This is not you, but watch out for this. They might say, Jim says drinking coffee increases the reaction time of all students, or decreases the reaction time of all students. If they've only done it on boys, then they can't apply their statement to all students and things like that. And if they've only done it on 15-year-olds, they can't say the same effect would happen on 7-year-olds or 20-year-olds. So just watch out when evaluating someone's results that they're never going to be able to, you're always going to be able to rip apart you're always going to be able to rip someone apart for their conclusions. So watch out for that. Yeah, same mass, gender, age, all of them type of things. Absolutely. Yeah, awesome. And always, it's better to use a computer. Why is it better to use a computer, guys? Why is a computer better than a human for doing the ruler drop? In fact, you don't use a computer for the ruler drop. It's not like a mechanical arm comes out. You use a touch screen where you tap the screen. It says tap the screen now and you go boom, like that. Why is that better? No human error. Good. Yeah, good. A computer is more precise, so can measure higher resolution, or you can say more precise, so it will be more accurate because of that, because it can go to more decimal places, more milliseconds, thousandths of a millisecond or whatever, thousandths of a second. But also the computer, you've got no thing where you can't read the body language of a computer. So watch out for that. You can't read the body language of the computer, okay? You don't know when the computer's going to ask you to tap the screen. All right, let's move on. Let's get off of that nervous system. Anyway, if you're rewatching this, you can have a go at that 6 marker and then you can pause it and use the mark scheme here to see how you did, okay? Because there's all the marks. All right. Just looking at the menstrual cycle, for those of you that have got the menstrual cycle coming up. So, those of you that have got the menstrual cycle coming up, does anyone know what hormone? Look how messed up this one is. Let's see if we can spot what these hormones are. Okay, I'll put my face over a little bit of it. Never mind. And we know what estrogen is there. Which hormone is X, guys? Y is LH. Well done, Sophie. It is LH. Luteinizing hormone around day 14 releases the egg. Ovulation, we call that. X is FSH. Well done, Sana. That's the one that ripens the egg. And what we should see is as estrogen over here, if I tag estrogen in as green, if I follow estrogen as green, when estrogen goes up, the FSH is going to start to drop as estrogen goes up. Negative feedback that estrogen inhibits the FSH, remember. What about Z? Anyone said Z? Adam has said progesterone. Well done, Adam. Progesterone. The job of progesterone to maintain the lining of the uterus, and if you're pregnant, that stays high. What's the evidence that this person is not pregnant? What's the evidence this girl, female, woman is not pregnant? Broski, inhibit means stops it. So when estrogen goes up here as estrogen goes up, the FSH is going to go down like that. So FSH is going down as estrogen goes up. It stops, estrogen stops FSH, inhibits. And the reason it stops it is because it doesn't want you to ripen any more eggs and waste them. Okay, is this woman pregnant? No, she's not. Well done, Gabriella. Her progesterone level falls, it drops. What does that mean? She's not pregnant. Okay, if she was pregnant, her progesterone level would stay high in order to maintain the uterus for implantation of the embryo, of the fertilized egg. Good. Nice. And that means her period starts again, Lucas. That's right. The lining of the uterus breaks down again and passes out the vagina during menstruation, which takes about 1 to 5 days. So coolio. Right. Let's move on. You guys smash that. Right. Some of you might have contraceptions coming up. Some of you might have contraceptives coming up in your exam. How do we know if they are pregnant or not? Oh, their progesterone levels will fall. If they're not pregnant, by day 28, their progesterone level will fall. Good. We've got the pill, Adam. Now, what does the pill contain? Estrogen, progesterone pills. Why does it contain estrogen and progesterone? Yeah, absolutely, Claire. If it's the combined pill, it's got them both. What do they inhibit? Yeah, well done, Adam. They stop FSH and LH. And why does that mean you can't get pregnant? If you inhibit or stop FSH and LH, why does that stop you getting pregnant? You need to be able to explain why it stops you getting pregnant. So, yeah, no eggs mature or release. Perfect answer, Miriam. Well done. No ovulation. Great keywords. Good keywords. Yeah. Yeah. You guys are all over that. Awesome. Right. You also need to know the alternatives because pills you can forget to take. So what are the alternatives to taking pills? Anybody know other hormone contraceptions other than the pill? There's like 3. Patch, awesome. So you guys, yeah, you're on that. You've got one called the hormone patch, releases them, might last a week. You've got the implant, which lasts like 3 years, goes under your skin, and you've got the injection, which lasts like 3 months. So you can have, oh, nice one, Gabriella. We got the plastic IUD. The plastic one releases hormones that inhibit FSH and LH. There is a copper IUD, which has spermicide that the copper is a natural sperm killer. I think it probably also releases does both. I think the copper one probably does both. But copper is a natural sperm killer, so that should kill the sperm. Now your barrier methods. What are the 3 barrier methods? The IUD goes into your uterus, Sana. So yeah, into the uterus. Yeah, into the uterus. Yeah, you got condom, diaphragm, which looks like a cup. You imagine your diaphragm under your lungs that domes up when you're breathing out. So it looks like literally the same as your muscle, the diaphragm that's under your lungs. So the condom, there's both a female and a male condom, and you can use spermicide with that as well. Awesome. And then other ways to avoid pregnancy. Somebody said, I can't spell it, abstinence. I think that might be it. Abstinence, that's basically not having sex. You can just write don't have sex. There is natural methods, which is don't have sex around day 14. So don't have sex around day 14. Why? Why is that not a very... Okay, Elliot. Yeah, that's not a very good method. Yeah, sterilization, right? Sterilization is permanent, so some people might say, yeah, that's a bit permanent. But you do have that. You can have tubes tied. And when I say tubes tied, got to be more specific. The fallopian tube or oviduct, it's called oviduct or fallopian tube in a female, and the sperm duct, which leads from the testes to the urethra and out the penis, is where the sperm goes. So you can have that tied, or you can have it cut. In very, very, very rare instances, the tubes untie themselves or fix themselves, which is very unusual, but they grow back. So somebody thinks, I've had my sperm tubes cut, I can't get a woman pregnant, me and my wife have had 5 kids, I've had enough, I've had my tubes cut, boom, 6th kid, because the tubes have regrown. And that can happen with a woman as well. The fallopian tubes can somehow reattach, but very rare. Yeah, it's hard to predict the cycle. Really, it's from, you can get an app nowadays, not dead easy, but you can get an app nowadays. So the first day of your period, you just put that in and it will tell you when you're ovulating. You know, it will say, right, ding, day 14, you're ovulating. So you'd know 12 days after your period, right, my egg's coming out anytime soon, so I'm likely to get pregnant. And this helps people, husband, wife, man, woman, boyfriend, girlfriend, who are planning on having a kid. It helps them time when to have sex to try and have a baby as well by doing that, by following the menstrual cycle. However, it's not a very good method, the natural method, because the egg could be released a week early or a week late. There could be circumstances which you can't control, and people can just do bad maths calculations. Right, let's have a look at the problem with, I've noticed Elliot says pull out method or Eggmaster pull out method. That's not really a method, and the reason is, from the moment a man puts his penis in someone's vagina, some sperm come out. So you can think, oh, I'll just put it in for 1 second and we won't get pregnant. That's not true. So sperm will actually come out the whole time, even you can't see it or whatever, so you can still get someone pregnant. And the younger you are, the more fertile you are, both for a female and a male. So men are at their most fertile, their sperm are at their absolute elite commando sperm when they're about 18 to like 21 or something, and women are the most fertile when they're 18 to like 25. So people practicing those methods, that's when you're most likely to get pregnant. So the pull-out method is not a recognized method of contraception. Right, you do need to know thyroxine metabolism. So thyroxine regulation, negative feedback. Okay, pituitary gland is in, where do we find the pituitary gland? Where would you find the pituitary gland? In the brain. Awesome, guys. Got a fly buzzing around me. Pituitary gland is in the brain. What does the pituitary gland release? It's known as the master gland. What does it release to go down to the thyroid gland in the neck? Does anyone know the name of the hormone it releases that goes down to the thyroid gland in the neck? It does stimulate thyroxine. Someone said TSH. Who said TSH? Well done, guys, who said TSH. The hormone TSH is thyroid stimulating hormone. Okay, thyroid stimulating hormone goes down to the thyroid gland, tells the thyroid, listen, your time, you got to release thyroxine, and the thyroid releases thyroxine. So, and then thyroxine, what does thyroxine do? Regulates metabolism. So it increases your metabolism, makes you burn energy and produce heat. So if you're stressed for your exam, what was your most stressful exam? During your most stressful exam, your body would have been releasing TSH, thyroid stimulating hormone, causing your thyroid gland to release thyroxine and increase the amount of glucose you're burning off. While you're stressing, you're releasing energy to keep warm and get you ready for, your body thinks you're going to have a fight. It doesn't know you're going to have an exam. We've evolved to fight. So increases your metabolism. Now we have an example of negative feedback here. So watch out for this if it comes up. When the thyroxine is released, it inhibits the pituitary gland. So you do need to know that by negative feedback. Thyroxine levels get high and inhibit the pituitary gland, so more TSH is not released. Now, you don't need to know what positive feedback is, but if we didn't have this negative feedback mechanism, the pituitary gland would keep stimulating thyroxine and it just go up, 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 up until you burn up all of your food and your metabolism is going super fast. So we need a system to bring it back to normal, and that's negative feedback. The thyroxine, when the level gets to a certain point, it stops the pituitary gland releasing more TSH, and then the thyroxine levels can lower back down. Okay. Yes, it does help with protein synthesis as well as with respiration. Cool. All right, looking at diabetes, etc., people, as this can come up with the endocrine system. So, type 1 diabetes, what's happening here to the person? How do we know this person has type 1 diabetes? Here's their blood glucose concentration. Why is their blood glucose gone up? Beth Hale, the pituitary gland doesn't tell the thyroid to stop producing. The thyroid doesn't produce TSH. The thyroid produces thyroxine. The pituitary gland produces TSH. Now, the thyroxine inhibits the pituitary gland once its level gets too high and stops TSH being released from the pituitary gland to stimulate the thyroid gland. So, yeah. Yeah, they just had a meal. Awesome. They've just eaten. And their blood sugar shouldn't go above 10. It shouldn't actually go above 7. Your blood sugar shouldn't really get too much above 7, so it's taking time. How do you think they've actually managed to bring it down? What's the problem? This person has diabetes 1. What have they had to do you think, to bring it back down? Yeah, the glucose is not going in the liver. They've had an injection. Well done, Elliot. Yep, they've had an injection, insulin injection. Yes, and that's brought it back down. That's the only way they can control it. Now, this bit of their body's working really well. What's happening here? It's gone too low. What's happening here? This bit's actually working well. So what's happened there? What after glucagon, Elliot? Yeah, glucagon's been released, Miriam, and it's moved their level. Look, it's gone back to normal. It's working perfectly fine. That's what should have happened after their meal. It shouldn't have got out of control and gone that high. So the glucagon's working well. What does the glucagon do to the, how does it increase your blood sugar level? Anyone remember how glucagon increases it? It is a negative feedback system, that's right. How does glucagon bring the sugar level back up? Converts, opposite. It's insulin converts glucose to glycogen. That's right, Ruby. The glucagon makes the glucose gone from the liver by converting glycogen back to glucose and raising that blood sugar level back. Okay, good. And just one more thing. What's the organ or gland that releases insulin and glucagon? Just to check you can remember which gland or organ. Pancreas. Awesome, you guys all over that. Right, I'm just going to look at one more and then we're going to move off of sugar and diabetes. We are going to get to meiosis. Those of you that are like, where's meiosis, mitosis, we are going to get to that, so no worries. Let's get rid of this. Alright, one more of these. Let me just put this image up. These are just taken from questions from different exam boards. Okay, which person, A or B? If you read the top of it, it will tell you anyway. But which person, A or B? Who's the diabetic? Yeah. Kind of all exam boards, Elliot. So all exam boards, really. Yeah, B's the diabetic. Look, their blood sugar level was high at rest, and their blood sugar level went crazy after they ate a meal and took a long time. If you look at person A, their blood sugar has gone up. That's normal, and it's come back down relatively quickly. In fact, within 1 hour, their blood sugar level's back to normal. But this is normal. your body is not a perfect machine. The blood sugar level, because it's regulated by hormones which need to be released and negative feedback mechanisms. So the blood has gone too low. Too much sugar has been put in the liver. Then we're releasing glucagon. We're getting it back to normal. So throughout the day, your blood sugar level every hour is doing that. But it should be within a narrow range if your hormones are working properly. It should be just doing that throughout the day. Okay, cool. Let's move on. Okay, DNA, guys. What do you guys know about DNA? What do we call each strand of DNA? Moving on to kind of inheritance, cell division, well, meiosis, sexual reproduction, selective breeding, and all that stuff now. Yeah, so nice, 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 nice. All right, good terms here, guys. You guys are coming out with some good terms, but just be careful. This here, it has a double helix shape. This is a polymer. It's actually made of two strands of polymer. There's two strands wound round each other in a double helix. That's the shape. What do we call a short section of DNA? What do you call a short section of DNA? Was meiosis in your paper one, Adam? Yeah, best to call it a gene. They are made of bases, triplet code and all that, but you do need to know it's a gene, and you have to know what does a gene code for. Yeah, codes for characteristics, codes for a protein. Yeah, specific sequence of amino acids. That's perfect. So now, if you get a change in your DNA, sometimes DNA changes and it's perfectly normal. It happens all the time. We call that a mutation, and that can lead to really good proteins being made, useful proteins. Okay, now, 1 second. We've got some Edexcel people in the house. So, yeah, and a triplet code of bases codes for an amino acid. So, a specific amino acid. But let me just check on Edexcel people. Yeah, some of this is paper one for you. I will be doing ecosystems as well, which is relevant to you. But I'm going to be doing a bit of inheritance. So if you've done Edexcel, if you're doing Edexcel, you might, yeah, I'm going to be doing something that you've already done for the next like 20 minutes. Okay, feel free to duck in and out of the live if you need to. I will be doing ecology and stuff later on in the live. Yeah, so for Edexcel people, this was in paper one, but okay. All right. Do you guys know, if you're doing AQA or OCR, do you know what is called when the egg cell and sperm cell join together? Okay, Lucas, it's fertilization. Beth, you're right. The new cell will be diploid. So that was cool that you said that. The new cell is going to be diploid. What does that mean that the sperm and egg cell are? Are they diploid? Yeah, Bev, that's right. They're haploid. And what does that actually mean? You might be sitting there, if you're someone sitting there thinking, what the hell does haploid and diploid mean? What does haploid mean and what does diploid mean? Anyone know what haploid means and diploid means? Yeah, there's 23 chromosomes in the sperm, 23 in the egg. But remember, okay, guys, Yeah, Lucas, I prefer the half the chromosomes, half the adult number or half the normal number of chromosomes. You should say 23 chromosomes, but remember, if they're going to be mean to you tomorrow in the exam, they might give you some creature you've never heard of, some weird fungi or a moss or some weird kind of worm, and they'll tell you a different chromosome number, but you'll still have to recognize that haploid is half, and that means it's the gametes, it's the sex cells, and they come together to make an adult cell. So what type of cell division, will have gone from here to here? Now think about this carefully. If these are diploid and then they're going to turn into the embryo, what's the type of cell division there? Would that be mitosis or meiosis? It's mitosis, Gabriella. Well done. Well done. It's mitosis. You can be fooled, and don't be fooled. Just because you see sperm and egg, it doesn't mean it's meiosis. Yeah, there's haploid in the diagram, but once we go diploid and we're just copying the cells, it's mitosis. We're going to look at that in a second, but we're just going to quickly run through sexual versus asexual. So the three facts about sexual. What makes something? This is sexual reproduction. Humans have sexual reproduction. Plants do. Well, they do both. Some do sexual, some do asexual. So, what do we need to know? If you get a 3 marker, what is sexual reproduction? What have you got to say? Yeah, 2 haploid cells joining together, egg master, become diploid. That's right. So, sexual reproduction. We get fusion of sex cells like that. So we get fusion of sex cells. You can say gametes join together. It involves gametes. How many parents? How many parents in sexual reproduction? 2 parents. Fusion of gametes or sex cells, you can say. How many daughter cells? Well, yeah, that's really going on to mitosis and meiosis, but fusion of sex cells, 2 parents, and we can just say offspring non-identical. It's better if you can say genetically non-identical. So genetically different or genetically non-identical. And then asexual is the opposite of that. There's no sex cells. So for asexual, no sex cells, no fusion of gametes, 1 parent, and the offspring are genetically identical. Okay, cool. Yeah, and when we make our gametes, it involves meiosis. So the making of the haploid cells in the testicles of a man or the ovaries of a woman involves meiosis. Everything else is mitosis. With asexual, for those of you that are doing triple, you need to know binary fission. Bacteria reproduce asexually, but by binary fission. But if you're not doing triple, you don't need to worry about that. Now, we've got a question here before we move on. A gardener wanted to produce a new variety of onion. Explain why sexual reproduction can make a new variety of onion. Why can sexual reproduction produce a new type of onion? Why do you not look like your brother or sister? Mixing of genetic information leads to variation. Yeah, we got genetically different offspring. Mutations, Sophie, there can be mutations, but generally sexual reproduction, we've got the random, we don't know which sperm is joining with which egg, so it's random fertilization of gametes, and we've got meiosis also creates variation, because when we do meiosis we only put half the chromosomes in, unlike mitosis where it's the full set. So you can say on the answer scheme to this, you would say meiosis can create variation, fusion of gametes creates variation. Yes, mutations can happen in asexual reproduction as well. Mutations happen in both, so they're not specific to sexual reproduction. Okay, right, let's look at meiosis. For those of you that hate this topic, there is meiosis here. Okay. So just check out meiosis. If I tell you that this weighs alright guys, for those of you that need to know about meiosis, how many cell divisions are in meiosis? By the way, if you could do me a favor as well and click out the chat and hit the like, that would be awesome as well, because that will help get it out to more people. Nice. Yeah, there's 2 cell divisions. So in the first cell division, notice the X-shaped chromosomes. They form an X at the start of meiosis, like that. Notice in the first division, the X's haven't been torn apart. In the second division, the X's are torn apart. Cheers. There's 2 divisions. Okay, but we get 4 daughter cells. So if you take 1, you multiply it by 2, you get 2, then you multiply it again, you get 4. So basically, they're dividing. Now, if I tell you this is something they like to ask. If I say the chromosomes at the start here, before meiosis, weigh 10 picograms, how much do you think the chromosomes weigh here in this one when they form X shapes? How many picograms do the chromosomes weigh here? You might say to me, what's a picogram? It's a unit of mass. How much would the picograms weigh where I put that arrow? What do you think? Thanks, Adam. That's right, Ruby. Ruby's saying 20. Look what's happened. They've doubled, right, Dylan? They've doubled to 20 picograms. Right. Now, you might get confused here. Think, okay, how much then would the chromosomes in the daughter cells weigh? So let's check out the second lot of daughter cells, the final daughter cells. How much would they weigh? How much do you think the daughter cells weigh? Look at the daughters, the final 4, compare them to the parents. The parents was 10 picograms of mass. How much would each daughter be? 5. Well done, Manny. So, 5 picograms. If you're confused, just think it's going to be half the parents, the first cell. Because it's meiosis, they end up with half the chromosomes, so they're going to weigh half. Why did they double in the middle? Well, they doubled in the middle because they're going to split twice. So we're going to go 10 times 2 is 20, divided by 2 is 10, divide again is 5. So we've divided it twice. Now, so watch out for that. They sometimes love to bust out this whole picogram stuff. So yeah, where they start talking about mass, and they're so light they're using a really small unit, which is very, very small. Also, nice. Right, let's move on. So that's meiosis. Mitosis only has 1 division. For those of you that have mitosis, I don't think it will come up, but mitosis has 1 division, 2 daughter cells, the same mass of chromosomes. The daughter cells are identical. These are not identical. Look at them. They're not identical to each other, and it's impossible for them to be identical to the parent because they haven't even got the same amount of chromosomes. So they're not identical to each other, and blah blah blah. Right. Let's go. Right. Here we go. Everybody's favorite, genetic diagrams. Right. Okay. Oh, by the way, you guys need to know the symptoms of cystic fibrosis. Potentially, if you're doing AQA, you need to know the symptoms of cystic fibrosis. Anybody know the symptoms? Anyone know the symptoms of cystic fibrosis? Yeah, it is of the cell membranes. Good. Mucus. It is a disease of the cell membranes. Mucus in respiratory system. You've got buildup of mucus in the pancreas as well. Cool. Now, next thing, the main thing that you need to know about cystic fibrosis. Cystic fibrosis is a recessive disorder. Right, guys, who can tell me the definition? You may well be asked, what is a recessive disorder? What is a dominant disorder? So what does it mean if it's a recessive allele or recessive disorder? Polydactyl is the other one. I'll go over that. So yeah, difficulty breathing, difficulty digesting. Nice. Yeah. Polydactyl. It's when you have an extra digit on either your feet or your hands. Yeah, so when you say only one is needed, we won't say dominant is most likely. No, dominant's definitely not most likely. So, recessive allele, you can say you only need one allele to get the characteristic, or you only need one allele for the disease or the disorder to be expressed in the phenotype, which is the physical observable characteristic. I like that, Ruby. Only expressed if it's homozygous for the genotype. If I say, I'll do dominant after because I'll do polydactyl. Right, now let's give you a little question on this. So Gary has, Gary and Sally, let's see if you guys can work this out. Gary and Sally do not have cystic fibrosis. However, their first child does. What is the probability of their second child having cystic fibrosis? So what's the probability of their second child? You'll probably be asked to use a genetic diagram, and it'll be like 4 marks. Like that, you guys. And if you have to use a genetic diagram, so what you're going to do, this. You're going to shade out the first box. I'm going to use F. Why do I know that Sally and Gary have got a big F and a little f? Why do I know that they must be carrying the cystic fibrosis recessive? Why do I know that? Well done, Amna. Why do I know? Alright guys, how do we know that Sal they don't have the condition, but because one of their child did, they both must be carrying the recessive one. So you've got to do a bit of detective work, and then you do the cross like this: big F, big F, big F, little f. Now in the exam, I just want to point out, you always put the big letter first. So even though it looks like, hey, I'm doing little f and big F, that would be his sperm. You don't need to draw little tails to make sperm. That's just me being silly. But if this was Gary, his sperm have a 50-50 chance which one gets there, but you put the big F first, and then we got little f and little f. So well done guys, 25%. Please, 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 if you're going to do a Punnett square, circle. The examiner may think, and he'll take a mark off if you don't. If you don't do this, he's going to take a mark off, or she's going to take a mark off. Don't want to be sexist here. Why do I have to circle little f, and point out that that is the person with cystic fibrosis? Okay, if you don't do that, the examiner might say, listen, I don't know if this kid thinks this is cystic fibrosis. They might say to you, hey, maybe they think that's the 25%, the two dominants. So you need to make it really clear that you understand the phenotypes. Phenotype means the characteristic. So you're going to say little f, cystic fibrosis, and then you can write normal, normal, normal on the others. By the way, obviously normal is, you can say without the disorder is a better way to say it, like rather than normal. People with cystic fibrosis are normal. Maybe I should put healthy, but healthy on those three, and this person has the disorder. Okay. Okay. Well done, guys. All good. Don't be picking on Sophie. Sophie's a superstar, people. Right, now. You guys were really good at that. I have got other questions on that, which I'll go on later for people that are still hanging about. Right, what's this all about? Yay. Yeah, no, Sophie's a hero. She's actually got a lot of people to come along to these lives, so awesome. Right, guys. Why does a zebra have stripes? 4 marks. Yeah, I'm going to do ecosystems. I'm going to go on to ecosystems in a little bit. Why does a zebra have stripes? Nice. Mutation. That's going to get you 1 mark. What do we need to say? We're going to go mutations. There's a specific answer. And I can change this question in lots of different ways, and the answer will be the same every single time. And this is about natural selection. So, yeah, the problem with saying that they're adapted to the environment, and they're going to want you to talk about natural selection here in the explanation. So the problem with saying they're adapted to the environment, it makes you feel like the zebra had a choice in it. The zebra didn't have a choice in it. Yeah, that's right, Manny. It is natural selection, and it starts with a mutation. What do mutations cause? They give us mutations cause variation. You can write that if you like. I'm going to write 6 things. You only need 4. Beneficial characteristic. You are not expected, by the way, to be able to explain why having stripes is a beneficial characteristic. They may ask you to suggest it makes them harder to catch. It actually, you don't need to know what it does, but it actually confuses predators when a lion is trying to jump on a zebra and it can just see 20 zebras with black and white stripes everywhere. It gets really confused. And so because of that, if you have the black and white stripes, you're less likely to be caught and eaten. We don't need to say that. We just need to stick to the bullet points. Mutation, variation, beneficial characteristic, more likely to survive. You are allowed to say survival of the fittest here, if you prefer to say survival of the fittest. So more likely to survive. If you're more likely to survive, what are you likely to be able to do if you can survive? Well done, Sophie. More likely to reproduce. Pass on beneficial characteristics. Well done, Ruby. And those beneficial characteristics are like genes. So you can say pass on beneficial characteristic or say pass on genes. Who do you pass the genes on to? Nice one, Gabriella. To offspring. So you pass the genes on to your offspring, and then if we want, we can say this repeats many times. So it will have taken zebras maybe 100,000 years, sometimes only 10,000 years, before the stripes evolved by natural selection, but that's it. Right, why have I got a shark on there? So, shark on there because... what could they ask you about the shark? They could say, why is a great white shark grey at the top? And you want to say camouflage. Yeah, that's right. But you need to come out with these points. If it's a 5 marker or 6 marker, you need to say, right, Grey on top because some were grey on top, some weren't, because of mutations cause variation. Okay. It was beneficial to be grey on top, camouflages them. Their prey you can't see a shark. If you look down and it's dark, the shark is dark, you won't be able to tell it's under you. So makes it more likely to survive, more likely to reproduce, pass on that characteristic to its offspring, and that's why sharks are all dark on top like that. Okay, I've got bacteria here. You guys need to know. You guys need to know about antibiotic resistance. So it's the exact same answer for antibiotic resistance. Okay, exact same answer for antibiotic resistance. Okay, they just reproduce asexually or by binary fission. So this is a bacteria here that has been lucky enough because of a mutation to have an antibiotic resistance gene. I come and take it makes it more likely to survive when we go and give you some antibiotics. All these bad boys die. This guy goes, oh, fancy that. I've got a resistant gene. And survives, reproduces by asexual reproduction, passes the gene on to the offspring by binary fission. And now the offspring are all resistant. And antibiotics have really helped us prove evolution. Okay, right. Nice. Decomposers. Yeah. I'm going to get onto all of that. Someone asked earlier, what is a species? I don't know if they're still around. What is a species, guys? How do we know two cats are of the same species, or two dogs are of the same species, or two humans are of the same species? Brilliant answer there, Gabrielia. Oh God, I can't even speak. They can mate and have fertile offspring. Okay, so mate. The two key words here are mate and fertile offspring. It's one thing to be able to have sex with something. You could, a dog could have sex with a chicken. Doesn't mean they can produce any offspring. But a dog and a wolf might be able to have a baby, but that baby will not be fertile. It won't be able to have its own children. So a dog and a wolf are not the same species, but they have a common ancestor. So once upon a time, they came from a similar species. Okay, so right. So the definition is you can have sexual reproduction, mate, and produce fertile offspring. That's the definition if you're the same species. If you're not the same species, you'll produce a hybrid which cannot have its own kids. And it's a two marker really if you're of the same species. Now we need to look at speciation. How does one species become two? So for example, where did polar bears come from? Well, once upon a time there was an ancestor of the brown bear and the polar bear. And what happened is they got separated by ice and mountains. And because of that, they could no longer have sex. The two bear populations, they were both the same species. They were both the same bears. Now, over 10,000 years of being separated by mountains, natural selection occurred differently depending on the environment. Polar bears were in the cold, the brown bears were in the slightly warmer environment, and so nature chose different mutations to survive, and therefore when they re-meet they can't have sex and produce fertile offspring. So polar bears and grizzly bears have a growler bear which can't have its own kids. So the main key points here for speciation are population becomes reproductively isolated. Okay, so population becomes reproductively isolated, can't get to each other. Natural selection occurs differently as their environments are different. After a long time, they can no longer breed and produce fertile offspring. So after a long time, they can't produce fertile offspring. Any questions about that, guys? No, this is in combined. Yeah, so speciation is quite simple. If you take a population, if you take a group of people and you say you can no longer, like if you take, I give this example, if a group of humans went to live on Mars, we forgot how to travel to Mars, we lost the technology, we didn't have any more spaceships, 10,000 years later, their bodies would have changed by natural selection. Different mutations will be favored on Mars, which means if we then refound the technology to make spaceships and we met up again, Earthlings and Martians, we could probably still have sexual intercourse, but we wouldn't produce fertile offspring. We'd no longer be the same species. We'd now become two different ones. So how did the original ancestor, by the way, the original ancestor would be extinct then, but how did the original ancestor of dogs and wolves, how did dogs, whatever it was, call it species X, split into wolves and dogs? Well, humans started to, one theory might be that humans started to take wolves in and keep the tamest ones and the nicest ones and leave the wolves, and so they no longer could have sex with each other, and over thousands of years they became two different species. So basically that's what speciation is. It's how one, and you'll need to know the difference. If you get a 6 marker, you may have to talk about natural selection and speciation. So they're kind of similar. They might give you a clue normally and say, explain how a shark developed a large fin by natural selection. Great. Explain how a bat developed sonar by natural selection. Explain how a bird developed wings by natural selection. Then you know to go into that whole thing, mutation, beneficial characteristics, reproduction, pass genes on to the offspring. You can go into that whole thing, repeating the process over and over again. But if it says, how did this one species become two species? Now it's speciation. You need to talk about that too. Okay? So, yeah, that's right. We're nearly done with this. I'm nearly done with this. So, you do need to know extinction. So, for extinction, what things can cause a species to no longer be in existence? Extinction is when there's no members of the species left on the earth, dead as a dodo. The dodos became extinct. So what can wipe a species out? You need to be able to name 4 things that can wipe a species out. Yeah, new diseases. What else can wipe a species out other than disease? Good one, guys. New diseases. Catastrophic event, volcano, earthquake, asteroid, apparently wiped dinosaurs out. New competition, Gabriella, that's right. So the grey squirrel came over from America to England. It's bigger, it's stronger. It's like beating up the English red squirrel, British red squirrel. And the British red squirrel is a vegetarian. The grey squirrel eats meat, eats vegetables, so it could out-resource them, gather more food, live in its trees, beat up the red squirrel, kick it out. It has more children when it has some kids. It has 5, 6 kids, so it outcompeted the red squirrel. So the red squirrel is now a protected species. You won't see red squirrels about in the park. You just get the American grey. So that's new competition, new predators. If we get a fox or something, a new species that comes and can eat something. So new predators, new competition, new diseases. Great, awesome. Right, let's move on. Selective breeding. Sudden change in the environment. Yeah, that's right, Sana. If there's a big change in the environment like global warming, that's going to create loads of extinctions because it doesn't give time for natural selection to occur. If there's not enough time for natural selection for a mutation to happen that gives some members of the population the ability to survive a change in the environment, then they're all going to die. So there needs to be enough time. When the change environments are slow, there's enough time for mutations and some members to survive. Right, selective breeding. 3 points. You need to know the 3 basic points of selective breeding. I want to get a cow, right, that produces loads of milk for my shop or my farm. Tell me how. I want to breed a cow that makes loads and loads of milk. What have I got to do, guys? So what are we going to do? Nice. So if they give you an animal, it's different to a plant or a bacteria or a crop. If it's an animal, you get 1 mark for saying choose parents, plural, with desired characteristics. The characteristics will be in the question. They'll say like cow that makes lots of meat, chicken that doesn't fly, I don't know, whatever, but they're going to give you a characteristic. So they'll tell you the desired characteristic, but choose the parents with desired characteristic, breed them. So that means get them to reproduce. So that's 2 marks. Then what do we do with the offspring who has said 2 cows who produce a lot of milk, desired characteristic, mate the offspring, then repeat. Yeah. So we then choose the offspring with the highest milk production. Well done, Sana. Yeah. So choose offspring with best characteristics and breed again. Repeat many times for many generations, if you like. And that's selective breeding. Watch out. That's nice and easy. Boom, you're going to smash the marks when you get like an animal one. But it's much harder if I give you this question. If I say this, I want corn. Tell me how I breed corn that are resistant to a disease. How do we breed corn that are resistant? It's not actually that much harder, but how would you breed corn to make them resistant to a specific disease? You can do genetic engineering, but by selective breeding. Yeah, you can do genetic engineering, but we can do it by selective breeding as well. So we can just say the same thing. We're just going to say breed. Instead of saying parents, you don't need to say parents here. So you just say breed corn with characteristics of resistance to whatever they say in the question, and then you'd say the exact same thing, repeat with the offspring, blah 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 that we've done there. Choose the offspring with the best resistance to the disease and repeat many times. So you can do it. We can do this with selective breeding, guys. We don't have to do genetic engineering, but that's a really great point, because let's just hit the genetic engineering quickly before we move on to ecology. So well, actually got a few other things to do first. Genetic engineering. So you can do it by selective breeding, so don't go into genetic engineering first of all. So genetic engineering, you guys should know, what do I use? So genetic engineering, if I have one plant and I want to take a gene out of it and put it in another plant, what do I use to cut a gene out? Let's say here is some DNA. Let's say this is the gene that I want. What do I do? Brilliant. Now, depending on your exam board, if you're an GCSE person, you need to know they're called restriction enzymes. But you can just say use enzymes to cut. If you're doing AQA, use enzymes to cut out DNA. Then use same enzyme to cut DNA. Yeah. Use same enzyme to cut DNA of the organism. So like, say I'm trying to insert this gene into a carrot, so I'll use the same enzyme to cut the DNA of the organism. And you use another enzyme. There's actually two enzymes used here. Use another enzyme to stick DNA in the organism's DNA. So you can use plasmids if you're trying to make insulin and stuff like that. Yeah, quadrat. A quadrat are you talking about? Yeah, you can insert into a plasmid. Yeah, you can transfer by plasmid or vector. Absolutely, yeah. Yeah. Nice. We often do this. This is how we make insulin. So everybody who's got a diabetic member in their family or something, we use, that's how they get their insulin. We cut the insulin gene out of a healthy human pancreas. Just take one cell of someone's pancreas, they're going to grow a new one, they won't miss it. We cut out the insulin gene using enzymes, and then we cut the plasmid of a bacteria, that's the circular DNA, using the same enzyme, so it makes the exact same shaped jigsaw cut, and then we can insert the gene using a different enzyme that glues it in called ligase enzyme. And then we can use a virus to take that plasmid, and because viruses are 1,000 times smaller than bacteria, to go back into the bacteria and deliver the DNA. We then call it a transformed organism. And we call the DNA recombinant DNA for those of you that are doing IGCSE. And that organism will now have the new desired characteristics. You can use a plasmid to get it into... Yeah, you can use a plasmid as a vector to deliver the gene from... Say I wanted to make orange peas. Peas are green. You've all had peas for dinner at some point in something. Peas are green. Say I wanted to make orange peas. So I take the gene from a carrot's DNA and I cut it using an enzyme. I then take that gene, I take the same enzymes and I cut the DNA of the carrot. But in order to... Some people say, oh, you need to use a plasmid. You can sometimes, but if I'm just doing a straight swap, I don't need to. I can take the gene and I can insert it straight into the pea's DNA using other enzymes to stick it in. In that situation, I don't need to use a plasmid or a virus as a vector to carry it in, but sometimes you can do. No, not by plants don't have blood or immune system. So if you're doing genetic engineering with plants or bacteria, they ain't rejecting nothing. They don't have white blood cells and they don't make antibodies. And if you're inserting a gene into a human, which is not allowed genetic engineering with humans, but if you were doing, we do it with mice and stuff, but if you were doing a genetic engineering with mammals that have white blood cells and antibodies like animals do, plants don't have that, bacteria don't have that. They don't have blood, so they don't have white blood cells, they can't make antibodies, memory cells and all that stuff. But yeah, so they won't have rejection. But in humans, you're putting the gene inside the cell, so the white blood cell doesn't know that you've done that. The white blood cell can only can't respond to that. It responds to antigens on the cell membrane. So anyway, zinging on, this is just something getting right near the end of this unit now. These are the 3 types of fossils. So I've got 3 pictures here. What are the 3 types of fossils for people that need to know about fossils? Miriam, gene therapy is where we try to change people's faulty genes, their faulty alleles. If you had cystic fibrosis, I would target the lungs, which is where the mucus is made, and the pancreas, and I would try and get into those cells and replace the cystic fibrosis faulty gene with a healthy gene that doesn't produce too much mucus. The problem with doing it in an adult like me, say I had cystic fibrosis, you couldn't do it to all my cells in my lungs. My lung is made of trillions of cells, well, billions if not trillions of cells. How are you going to cut out the nucleus? You could do some, and so at the moment it has very limited use. However, if I'm going to have a child, this is a theoretical gene therapy. If I'm going to have a child, we can't do this yet, but in theory we could go into my sperm cell, or we take my sperm and my wife's egg, join them together, and then when it's one cell, there's only one piece of DNA I need to get into and cut out the gene and replace it with a healthy gene, and then my child won't have cystic fibrosis. So that's not allowed yet, but that will be the future use of it, being able to go into the fertilized egg when you've only got one nucleus to cut into. But to try and change all the cells in my lungs, it just ain't going to happen. Okay. Yeah, so we got 3 types of fossil here. We've got mineral replacement, you need to know about for tomorrow's exam. Mineral replacement. Okay. This is where, so that dinosaur where the two people are cleaning the dinosaur, it looks like it's bones. It's not bones. That's not dinosaur bones. That is where the calcium from their bones has been displaced in a chemical reaction with other minerals over millions of years, leaving behind a hard, what looks like bones, fossil. So that's really good. Downside to that is we don't know the skin color, we don't know if it has feathers, we don't know the eye color. None of the soft parts are left. They've all decayed by microorganisms. This is in combined and triple. I can cover triple, anything on triple as well. Everything I've covered so far is coming up in triple as well. So, but this is also in combined. This is known as impression or casting. This is where the organism leaves a print of itself in wet mud, and over thousands, well, that hardens into clay, and over thousands of years we have a footprint, a trace, a body outline of the animal. There's none of the organism left, but we can tell things about the organism, how many legs it had, did it have a shell or an exoskeleton. Okay, if you're doing AQA, you need to know the 3 different types of fossils comes up all the time. But it depends if you're doing EdXL or OCR. And then you have preservation, which is how a fossil is formed. So preservation is when the organism is kept. Got a mosquito inside this amber. People have amber as a jewel. It's the sap that comes out of a tree, the sugary sap that comes out of a tree. And when it hardens, if there's an animal trapped in it like this mosquito, it won't decompose. And you really have to know that it can't decompose in preservation because oxygen can't get to it. Or if it's a mammal, like a woolly mammoth, and it's frozen in ice, the temperature is too low for the enzymes of microorganisms to decompose. Decomposition can't happen. Yeah, so you do need to know you've got mineral replacement, where the harder objects like bones and teeth are replaced and they form a hard substance or sediment, like Gabriella said there, leaving behind what looks like a skeleton. That's really cool, but you don't get the soft parts. Then you've got casts and impressions, which are where footprints, a worm could wriggle through mud and leave a path. The roots of a plant could grow through the mud. The plant rots away but leaves behind an impression in the clay of what it looked like. And then the best type of fossil ever is the preservation, where we find something like a woolly mammoth frozen in the ice and we get its skin color, hair color, eyes, bones. It's all left there. And the idea is that one of the 3 methods of decay is missing: moisture, oxygen, or temperature, so the organism can't decay. Okay, yeah, that's right, Sana. There's something stuck in amber, so oxygen can't get to that mosquito, and the bacteria cannot respire and break it down by decomposition. AQA practical, yes, Misa, Misa. My pronunciation's rubbish. You have to forgive me. Are you doing combined or triple? If you're doing combined, the only practicals they can ask you are reaction rate and quadrats, estimating population size, or the transect for looking at distribution. If you are doing triple, you have the decay practical as well. So really, you've got the reaction rate practical, the ruler drop. You've got the random quadrat when you're estimating population size. And then you've got the line transect, where you're looking at the distribution. But if you're doing triple, then you need to do the decay practical as well. Yeah, you won't need the decay practical though. Right. Yeah, for those of you that do combine, you don't need the decay practical. Carl, quick thing before moving on to ecology anyway. Carl Linnaeus. Carl Linnaeus, he decided to classify all living things into king, prawn, curry, fat, greasy sausages. So kingdom, phylum. You don't need to know any of these. You just need to know this order. Kingdom, phylum. What is it? Class. Oh my gosh. Order. Kingdom, phylum, class, order. Oh my gosh. I'm losing. Yay! That's what Carl Linnaeus did. You need to know kingdom, phylum, class, order, family, genus, species. You need to know that order. They often do like missing the gaps kind of thing. You need to know that as we go down, we become more closely related. So, for instance, chimpanzees are in the same family as us. You're in the same family as a chimpanzee. You might not like it, but they're in your family. But they're not in your genus. There's nothing left in your genus. All homos are dead. Homo habilis, Homo erectus, all the other homos that wandered the earth are all dead now. Maybe some people think thanks to us, because you know what humans are like, Homo sapien. We like to kill everything. So we've got nothing of our genus left alive. We're the only Homo sapien. Yeah, there's no one else in our genus on the earth. So, but we do share a same family with other organisms. And you share the same kingdom as all animals. So you are related to insects. You are related to fish, as they're in the same kingdom as you, because you're part of the animal kingdom. Now, so you should be all familiar with this. The 5 kingdoms, just they like to call them funky animalia, but just animal, and plantae, but you can just say plant. There's 5 kingdoms: animal, plant, fungi, protist, and bacteria. So originally, for 300 years, up until 1990, so 30 years ago, from 1700 to 1990, it's nearly 300 years, 290 years, roughly, we've always separated all the organisms on the earth into animal, plant, fungi, protist, bacteria. And then you get more closely related as it's a hierarchy of separate groups. And as we go down the groups, organisms are more closely related, but there's no overlap between the groups. Now, Carl Woese, he came along down here, this dude. So you do need to know the name Carl Linnaeus. If you're doing AQA, you need to know about Carl Linnaeus. He's the guy that came up with the classification system. This one. Carl Woese, 1990s. He did RNA analysis. You can just say from the new information about chemicals or biochemical information, the study of DNA or RNA, he came up with, he noticed that bacteria were not all similar, and that some were very different than others. So he threw a spanner in the work. He basically said, look, I've looked at the DNA of different bacteria. I mean, what a dude. That's what he decided to do with his life, but he's famous now. So and he said, actually, bacteria, before you put them in a kingdom, there are 2 types of bacteria. Does anyone know the 2 types of bacteria? Yeah, RNAs in protein synthesis. Bacteria is not eukaryota. That's right. So he noticed that one was I need to move my camera because I've got my camera right over that. Move. Right, let's zing back here. Okay, so well done. People are saying archaea and modern bacteria. Nice. So you can, some people call that eubacteria, which is a bit dumb, but that's what some people call it. It's a legitimate one. You can just call it bacteria. And archaea is also called archaebacteria. It just means old bacteria. You need to know that archaebacteria are bacteria that are found in caves, bacteria that are found at the bottom of the sea and deep sea vents, and they're very ancient and very old. And he noticed that their DNA or their genetic code, their RNA, is very different than the modern bacteria. And the modern bacteria had quite a lot of different proteins and different chemicals. And he said, look, we got to split these up first. And then he said, well, although we say that, are you an animal or a plant? Okay, you guys are all animals. You're not plants. You're in the animal kingdom. He said, but hold on. Animals, plants, fungi, and protists are all related. They're all eukaryotes. So he called these the domains, the 3-domain system. So he said there's a level above kingdom. When you first discover an organism, you need to say, is it a eukaryote? Is it eubacteria? Is it archaea? If it's a eukaryote, then you go to the kingdom system and say, is it an animal? Is it a plant? Is it a fungi? Is it a protist? Then when you choose which one it is, you say, right, what phylum is it in? So it's a way of classifying. So he just added an extra layer, and it's called the domain system. The domains. And they are those 3 up here, guys. Anyway, cool. The domains are 3 things: eukaryote, eubacteria, and archaea. So you really just split the prokaryotes into eubacteria and archaea. You can call it true bacteria, absolutely. That's what I think they use in the book. Prokaryotes, really, true bacteria and archaea are both prokaryotes. Both don't have a nucleus, but he noticed a big difference in their genetic code, so he said they're not the same. Great. So what's this about? This is in a question where they're going to tell you about how new evidence leads us to change existing models. So every single thing you do in science today and in your B2 exam tomorrow, in 100 years, will be different. You would write all the answers you write that you get marks for tomorrow, you probably get crosses for in 100 years, because science is just models and they change. So this person has said, right, we're classifying it like this now, so this is how we're rocking now. Now, you do need to know about the binomial system. If you're doing AQA, you need to know about the binomial system. The articles normally aren't too hard. It's mainly comprehension, so you'll be totally fine. I think you'll be really fine with the article. Will be fine. Now, you need to know the binomial system. Okay, if you're a human, you guys are all human. So, as you're human, you're all eukaryotes. Everyone watching this is a eukaryote, I should hope, unless there's some clever plant or fungi out there. Everyone watching this is a eukaryote. That's your domain. Your kingdom is Animalia. Your phylum is Chordata, or vertebrate. Your class is Mammalia. And then we can go all the way down to your genus and species. Now, it'd be really awkward if I said, hi, my name's Tom. I'm a eukaryote. I'm Animalia. I'm Chordata, or vertebrate. I'm a mammal. It gets a bit long. So what we do is the binomial system just names every organism by the last 2 taxons or levels of classification. And that is, we say the genus and then the species. So we say genus, then species. All right. And you do need to know that the genus is always written with a capital letter and the species a lowercase letter. So it would be Homo, and then we are sapiens. So we'd be written like this: Homo sapiens. The species is lowercase. The genus, it starts with a capital letter. Okay? Cool. Right, just going to clear this, and I'm just going to do a quick example of something, and then, yeah, like so. Rubbish. What is that? Okay. Cool. Now, let's just move. Let's just move. Last thing here is, yeah, Ellie. yeah. I've kind of left the old ecosystems far behind. By the way, I did do a live on ecosystems. I'll put the link up, but I can go through ecosystems if you want, or anything else after this. I'll go through any questions that people want. Last thing here, this is an evolutionary tree for the great apes. All right, we belong over here, by the way. We're hominid. That's our either our order or family. I can't remember which one, but we're in there. So what do you need to know about evolutionary trees? I'm going to ask you a couple of questions and see who is on it with the evolutionary trees. So if you're doing AQA, got to know the evolutionary tree. So which 2 of the apes are most closely related? Which 2 apes on this tree are most closely related? This is quite a difficult one, but which two are the most closely related? Well done, Umna. It's the bonobos and the chimps. Nice one, guys. Now, some of you might have said, few of you might have been tempted for that. Anyone tempted for that? Okay, you might have looked and said, these are closest together. You got to follow the line back and go, actually, they're really quite far back. Like, if you get your ruler out, you can even measure it. They're a lot further away. These two dudes, the bonobos over here and the chimpanzees share a much more common ancestor. So bonobos and chimpanzees share a more common ancestor. Okay. No. In IGCSE, I don't think they're going to give you this. Now, which of these evolved first? So, anyone tell me which great ape was first to evolve? Which one came on the scene first? Anyone work out which of these must have evolved the first? Yay! Well done, KDP. It is orangutans. That's right. Yeah, well done, orangutans. So they evolved way back here. They split from this. But all of these organisms are related to this ancestor. And it goes all the way back to a single... So if you follow the theory of evolution, we all go back to one single cell that started on the earth. There was the very first cell. It wouldn't have looked anything like a eukaryotic cell. It wouldn't have had a nucleus. It wouldn't have had anything. It would go back to just a little bit of cytoplasm with a piece of DNA. We don't even have any idea what it was, but it was going to be simple. Probably something simple to those, like more like those archaea bacteria. But you can follow the ancestors back. Wherever these two lines meet is a common ancestor that is now extinct, doesn't exist because it's speciated by geographical or reproductive isolation into the two things that come off of it or more. Anyway, enough of the evolutionary trees. Right, what topics do people want to go over? I'm hold on. Whew. Right. So I can go over different topics. Just looking at what I was just gonna say. If I zing to yeah, I've kind of done homeostasis. If you rewind it back, you'll find homeostasis there. Yep. I'm just going to put a link up to an ecology one. I did just in case. Hold on one sec. Where's my ecology stream? Okay. Predator prey cycle. I don't know. Oh, that's me just waffling. Right, let me just copy this link. Oh my days, what is happening there? Copy. Alright guys, give me one sec. Now, in theory... Alright, listen, I just put a link up there. Can you see that link in blue there? That's to a live I did about 5 days ago on ecology. Yeah, but you can kind of watch that later. Your exam, if you don't have geography, you've got the luxury of being able to watch stuff in the morning tomorrow in the afternoon. So that's a link to do with ecology. If you're doing Edexcel, you need to know about plant structure, the xylem, the phloem, and phototropism, plant hormones. I've also, so that link there that I've put up will take you to ecology. And I've also got Yeah, homeostasis was done earlier on here. No, no, AQA combined. You don't have to know about xylem and phloem. I've gone over classification. Yeah, I've gone over classification a little bit with Carl Linnaeus and his systems. Um, what else? What else? What else? What else? So I've also got. I might just quickly for the EdXL people do a bit of. Let me very quickly. All right, Adam, I have a live on photosynthesis I did as well. So I've got one that's purely on photosynthesis you can look at um, on photosynthesis. I've got another live I did that is just covers everything in photosynthesis. So Nihat, micro propagation is so micro propagation is just when you take some tissue and you take some cells. Let me have a little look if how much detail you need, and you're going to grow that in a nutrient agar and make very quickly. It's quicker than you're going to make millions of identical copies of plants. But let me have a little look about micropropagation. So yeah, so if you're doing IGCSE, you need to take the tips, the root tip. So this will take 2 minutes. Root tips of plants. So they contain meristem tissue. Meristem tissue is the bit where they contain stem cells, basically. So you're going to take the root tips of plants. You can take the shoots, and you can take the shoots, or take shoots. I should say shoot tips. So if you want to grow loads and loads of plants, take the root tips and the shoot tips. Yeah, I can go over eutrophication. I can go over anything people want. I'll just quickly go over this one. Um, Now you need to sterilize the plants to kill any diseases, any microorganisms. So you're going to sterilize them, kill any microorganisms. Then you don't need to know the name, the callus, but they grow into something called a callus. So add growth hormones like auxins. So add growth hormones like auxins. Supply with nutrients. Once they become small plants, you can put them in pots. So what you tend to do, micropropagation, you need to know, is really good for guaranteeing genetically identical offspring. Okay, so you just take a root tip of a plant, you can chop it into so many pieces, and because you're only using cells, you can actually make millions. So it's better than cuttings. Cuttings is when you chop pieces off and then stick it in some soil or agar and you grow it from the cutting. But here you're using cells, so because you can make so many millions of cells from 1 centimeter of a root tip, you could make thousands and thousands of identical copies of the plant. And you're going to sterilize it first to make sure there's no bacteria or viruses, then add some growth hormones, some nutrients like glucose, amino acids, and then grow them. And once they become a little ball or a little lump, you can plant them out in pots. They call it a callus, but you don't need to know that for IGCSE. And that's micropropagation. Um, Tasha, are you doing? What are you doing? IGCSE or AQA? Eutrophication comes up in a little bit in yeah, peat bogs. Yeah, I'll go over peat bogs. Yeah, cuttings are bigger than explants. You're right, Niha, but that means that with cuttings, there's only so many cuttings you can take from the parent plant before you kill the parent plant. Whereas you can grow, yeah, if you take an explant, a small piece, it has far more cells in it. Like micropropagation is using the cells, so you're going to make it into really small pieces. A cutting's bigger, but yeah, you can't grow as many from cutting. Yeah, you probably don't have to do eutrophication, but just in case, let me go over water pollution for those of you that water pollution can come up. So for water pollution, if you're doing eutrophication, take 2 minutes to do eutrophication. So with eutrophication, okay, fertilizer or sewage. I mean, you might say to yourself, why is the farmer pooing on the field? But that's just because I haven't got time to draw a sewage plant or people's waste. So sewage plant, the farmers releasing some sewage, some waste. Okay. Just for those of you that have eutrophication, it's going to rain. This is going to wash the sewage and fertilizer into the lake via a process called leaching. What's going to happen then? We're going to get an algal bloom like that. We call this an algal bloom. That's going to then block this sunlight from reaching the plants that live inside the lake. Yeah, they look like mountains, but they're supposed to be plants. So, so producers in lake die as no sunlight. You can expand upon that. No sunlight, they can't absorb sunlight, so they can't do photosynthesis, they can't make glucose, they can't make proteins, whatever, they're going to die. What that means is no oxygen. If the producers aren't doing photosynthesis anymore, there's no oxygen, and that kills the fish. There also are microorganisms that feed on the algae. When the algae here die, the microorganisms use the oxygen in respiration and use it all up, and the lake becomes deoxygenated, no oxygen in there, and that kills the fish and kills everything eventually, and it becomes a dead lake with nothing living in it. It can happen to rivers and streams and any body of water. It's not going to happen to the sea, it's far too big for that to happen to the sea, but to closed bodies of water, streams, rivers, lakes, whatever, it can happen. Yeah, so rain, sewage. So what do you need to know for AQA combined? Do you need to know that level of detail? No. IGCSE you do, and maybe triple you do, but for combined, you just really need to know that when it rains, leaching's a good keyword to score you marks. Sewage and fertilizer can run into lakes and toxify, kill. You just need to know the result is the death of organisms in the lake. Toxic chemicals can run in there. Also, pesticides and herbicides that farmers spray on their plants can run in there, and they can kill things in the lake. And that's what you really need to know for if you're doing combined. If you're doing triple, you need to know a little bit more about how the leaching creates the algal bloom or the growth of algae, and they block the sunlight, they prevent photosynthesis, and that kills the fish and kills the plants. Decomposers use up the oxygen, and the producers can't produce oxygen, so it becomes deoxygenated. That's what you need to know if you're doing IGCSE or triple. But if you're not, you're doing combined, you just need to know that when it rains, fertilizers and sewage runs into the water and causes water pollution, and herbicides and pesticides run into the water, and they're toxic. They make the water toxic. Okay, right. Yeah. There's other streams as well, by the way. You can there's loads of other things. Yeah. Peat bogs. Someone said peat bogs. I don't know if um yeah, if you're doing AQA, you need to know about peat bogs. Okay, so okay, what's a peat bog? A peat bog. Okay, so peat bogs um, provide habitats for many species. So you guys need to know that peat bogs provide habitats for like loads and loads of species. Love to live in a peat bog, and if we destroy peat bogs, we're destroying the habitat for these species. But also, um, peat bogs are good fuels. And this is the reason that people are burning peat all over the world. It's a really good fuel. It's slow release of heat, really good, keeps you warm in the winter, better than burning wood, even better fuel than wood. So peat bogs are good fuels. People burn them, and this releases CO2. What's the problem with releasing carbon dioxide? What's the issue with releasing carbon dioxide? Well, carbon dioxide, of course, remember CO2 is a greenhouse gas. And then, yep, that's right, Bev. You want to link that to global warming. So, destroying peat bogs is going to increase the greenhouse gas and the global warming. And if you get a question on the greenhouse effects, that just think what's going on. If any of you have watched the news, what's going on at the moment? We've got forest fires, we've got floods, we've got a change in the distribution of organisms, migration patterns of birds. Birds normally fly to England at this time of year from Africa. It gets too hot in Africa in certain parts, so they chill here in England for our summer because our summer's cooler. Well, if we get global warming, they're going to need to go somewhere else. So it's going to change the migration patterns of animals. It's going to change the distribution of organisms. But back to peat bogs. Peat bogs provide habitats. Burning them releases CO2, so that's a bad idea. The other thing that us humans do with peat bogs, people use peat as fertilizer. So we use peat as fertilizer. Why? When it breaks down, microorganisms love it and they break it down, and it gives so many nitrates and other rare nutrients into the plants that the plants need, that your crops are going to grow brilliantly, or the flowers in your garden's going to grow excellent. People use peat as fertilizer. As it decays, you need to know that as it decays, microorganisms, decomposers, or bacteria or fungi release CO2. So again, that is locked in the peat. So basically, peat bogs are a great sink of CO2. Where's a lot of carbon dioxide is locked there. They've formed over hundreds of millions of years, so we can't remake them, and we're releasing all the CO2 from them, and that's causing global warming. So, yeah. Okay, so that's the thing about peat. Peat's also actually, we found a lot of we found humans from 10,000 years ago who've fallen into peat and been preserved. So like the amber, things don't decay when they fall in peat. And so, yeah. But we're destroying the peat, and it's equally bad. You need to know about deforestation. Chopping down the trees increases the carbon dioxide level by 3 ways. What are the 3 ways that carbon dioxide increases from chopping trees down? So the 3 ways, brilliant, like that one. Trees are a store of carbon. Photosynthesis can't happen. Brilliant. So photosynthesis removes carbon dioxide. So we're getting less removal of carbon dioxide. Trees lock up carbon dioxide, so we're having less carbon dioxide locked up. And then what do people do to the trees? That's right, Tasha. And then people combust the trees. They burn the trees, releasing carbon dioxide. Or fourth one, the trees decompose. You chop a tree down and you leave it just lying there. Bacteria break it down and decay releases carbon dioxide. So there's four things you can say there on how they increase global warming, deforestation. You do need to know the reasons why we chop down forests and trees. Yeah, agricultural uses. As the human population increases, we need more farmland, more agriculture to feed the people. So we have to chop the trees down to create farmland. So there's a conflict there. We don't want people to starve. We need to grow more food to feed people. So we have to chop the trees down for farming. We also have to chop the trees down to use to build houses and make room for people to live and build cities. So there's a conflict there going on. More demand for food. We need food security. Great word there, food security. And yeah, and also for making paper and fuel. People need to stay warm in the winter. Yeah, more demand for food as population increases. Great stuff, guys. I did say, hold on. Like I did do a live stream ages ago on photosynthesis, which I'm just going to quickly put in the chat. For those of you that need to do photosynthesis, I'm just going to quickly put this in the chat. A lot of people have done photosynthesis. Just talked about micropropagation a little bit. This is for photosynthesis there. If you need to do photosynthesis, that's a 1 hour live all on photosynthesis. Yeah, so Nihat, yeah, decomposers, Tasha, decomposers are bacteria and fungi, and you can link this to the carbon cycle, but yeah, decomposers break down organisms and return carbon dioxide to the air. Just be aware in the carbon cycle, carbon dioxide from the atmosphere goes into organisms by photosynthesis and then is turned into carbon compounds. Don't say that an animal then eats a plant and gets the carbon dioxide. It doesn't. It gets a carbon compound, i.e. glucose, starch, protein, amino acids, or fats. They're all carbon compounds that plants make. When we eat plants, we get those carbon compounds, but then we release the carbon as carbon dioxide in respiration back to the atmosphere, or we die, plants die and decompose, and then carbon dioxide is released back to the atmosphere while the bacteria and fungi feed on the carbon compounds and break them down. They release the carbon dioxide. Of course, humans have come along and done combustion and deforestation, and they all link to the carbon cycle. So chopping down the trees means less removal of carbon dioxide, and burning the trees means more combustion, more increase in carbon dioxide released. Yeah, oh, by the way, remember that plants respire too. Plants do respiration as well. So don't forget, when you're looking at a carbon cycle, you're looking to spot that as well. Nihat, sexual and asexual reproduction. Yes, you guys need to know the advantages. So let's just quickly talk about those. So, sexual reproduction, that's what humans do. There's advantages to it. And some plants do both. Raspberries do asexual reproduction. Strawberries do asexual reproduction. But guess what? They also do sexual reproduction. So they do both. So they're kind of like got a good advantage there. Sexual reproduction. Very good, Amna. The big advantage to sexual reproduction is variation in the offspring. Why is that an advantage? If your children, offspring in biology, don't look like you, don't have the same genes, then if a disease comes along that kills you, it might not necessarily kill your children because they have different genes. The disadvantage to asexual reproduction is a lack of variation. So, you're less quick to adapt to changes in the environment or survive diseases that can take out the whole population. So basically, sexual reproduction, more variation in offspring. That's the advantage. More likely to survive changes to the environment. However, like global warming, things that sexually reproduce more likely to survive because there's more variation. So if you look at humans, we've got people with black skin, we've got people with white skin, we've got a lot of variation. We've got tall people, short people, fat people, thin people, brown hair, blonde hair, blue eyes, brown eyes. We've got lots of variation, so the human race should survive changes to the environment. However, downside to sexual reproduction, you have to find a mate. Half of the human population is spending its time trying to find a mate to reproduce with and have children. That's the big focus of lots of energy wasted on it. Whatever species, if you're a dog, lots of energy wasted. If you're a plant, you have to produce a flower with pretty yellow and purple petals and say, "Come to me, bumblebee," and that uses loads and loads of energy, right? Lots of wasted energy. So that's a disadvantage. Lots of energy used attracting a mate. It takes longer to reproduce. However, asexual reproduction, disadvantage is less variation, which means you're less likely to adapt to changes in the environment. However, less energy used, as you do not need a mate. So you don't need someone to reproduce with, you're just copying yourself. So less variation, obviously as identical offspring. Less energy, as you do not need a mate, reproduce faster. So you can't really say one's better than the other. Like a bacteria in the environment, just because it's having so many, it's reproducing so fast, every 8 minutes it's having kids. It couldn't do that if it had to find another bacteria to mate up with. It had to do a special dance and a song. When you wake up in the morning and you hear the birds singing, a lot of those birds are male birds. They're trying to attract a mate. How much energy are they using every morning going, singing a little song, doing a little dance and trying to attract a mate? They're using a lot of energy. But the advantage is their children will have different genes, so more likely to survive a change in the environment. One parent in asexual, which means less is more energy efficient and it can happen faster. So they can reproduce faster and beat the competition. If I'm a strawberry plant and I'm producing runners are little other strawberries that are coming off of me, little shoots that come off and identical copies of me grow, then we can put our leaves up and outcompete all the ones that are waiting for bumblebees. We can outcompete them. And we can also, strawberry plants have flowers as well, so they're attracting mates as well. They're attracting bumblebees, so they're reproducing as well sexually. So they get the benefit of both. Yeah, that's right. If you've got less variation, you're less resistant to new pathogens. Yeah, Adam, you've got to know the chemical equation for photosynthesis. If you check out that live, it's about an hour long. So if you don't have geography tomorrow, you can watch that tomorrow. That goes into chemical equation for photosynthesis. It goes into all of the stuff you need to know, the limiting factors and everything like that. Yeah, what I want to do, let's have a quick, let me just, general, but you can just ask, one sec, oh, whoa, what's happened there, he says. Got some questions, so just zinged off of my Jamboard. I'm just gonna give you guys some questions. Yeah, I've got some other files there, as you could see. Right. All right. This is just on the endocrine system. Just got a few questions for some of you guys. So, Can I move that? Let's move that over there, make that a bit smaller. Right. Okay, guys. Just some questions just to see. Which organ system includes the gland shown in the figure above? And what about, yeah, endocrine, easy peasy pudding and pie. Which gland produces insulin? Which gland produces, yeah, C is, yeah, 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 yep. How do hormones travel from one gland to another? I think Amna said the bloodstream, that's right. Name two glands involved in human reproduction. Ovaries and testes, Amna again. Ovulation test kits help women know when they're most fertile. Ovulation test kits detect the hormone that stimulates ovulation. Which hormone would be detected around ovulation? The 14th day, which hormone goes high? That'll be LH. Yep, absolutely right. Okay. These are just some random questions for paper 2 tomorrow. Anyone know what's meant by biodiversity? Variation in a species. Doesn't it? It's not really just with one species, it's the number of different species in an environment. So if my garden has fox, rabbits, mice, and apple trees, that's 4 species. If your garden has 6 species, yours is more biodiverse. And that's what you have to know at GCSE. The more species in one area, the more biodiverse it is. Okay. And the more healthy that ecosystem is, the more likely it will survive changes in the environment. Right. So gaming, UC gaming, biodiversity is the number of species in a community or the number of different species in an ecosystem. So the more biodiverse, the more different species they are. You can actually look at, not at GCSE, but at A level, we look at biodiversity in terms of genetics as well. So we look at one group like humans, and we say, are humans biodiverse? We can look at the number of different genes they have. So you can look at genetic biodiversity. Humans are quite biodiverse. This is why we reject organs all the time, because we're not that similar to each other. Other species like cheetahs, you can take the heart of one cheetah and stick it in another cheetah and it won't get rejected. They're very similar. They don't have a lot of diversity. Anyway, one use of peat from peat bogs. What a peat, yeah, make compost like that. Nice. Explain the use of peat is harmful to the environment. 2 marks. Why is peat harmful to the environment? Burning peat releases CO2. Yeah, anybody got another one? So that's global warming. Why else is using peat harmful? Do not refer to biodiversity. Yeah, it's used as compost or fertilizer, produces CO2 when you burn it. It's also the habitat. It's also you're destroying a habitat. Beth, it said you're not allowed to use biodiversity in your answer, which is a bit of a bummer because they talked about it earlier. They wanted you to come up with a new keyword. So, yeah, it'll reduce the number of habitats. Yeah, it creates wasteland. Yeah. Okay, over here, guys. Punnett square. Who's on the? I need to move my. No, yeah, Punnett square, guys. Let's Punnett square this up. Dominant, they're using T. Just because it's cystic fibrosis, T's a great letter to use because capital T and little t look different, so you can't mess up. Always make your noughts and crosses board and colour out the first one because you don't need it. A man and a woman do not have CF. The man and woman have a child with CF. Hey, this is the same as the one I did earlier. 25%. Did this one earlier. So, guys, I'm not going to do this one because I did this exact one earlier in the live, right at the start of the live. That's right, it's 25%. I'm not going to bother doing it because we did it at the start of the live, and it's going to be like that. And you know they had to have the recessive allele. I say I'm not going to bother doing it, and then I go ahead and do it. Now, why is it worth 4 marks? Let me just see. Make sure you put the capital there. Oh, how did I mess that up? Spot the deliberate mistake, guys. Right. You will only get 3 marks for this. This is only worth 3 marks, even if I say 0.25. What have I not done? I've missed a mark. I missed the mark here. I missed the phenotypes. Please, please, please remember phenotypes. Ooh, I need to move my camera. Remember the phenotypes, yeah? Yeah, put the ring. That's it. Ring that one. You point out that that is that. Because the examiner might say, well, big T is 25%. You thought it was that. I'm not giving you the mark. Even better if you can just write, this person has cystic fibrosis. Now the examiner knows, yeah, this guy, this student knows that's the one with cystic fibrosis, rather than just isn't sure. Right, let's check out another one. Okay, 6 marker. Who's ready for the 6 marker? Right, person. Let's have a look at this. So, person 7 and 8 plan to have another child. Okay. These are called pedigree diagrams, and they can be quite hard. So persons 7 and 8 are going to have a child. Determine the probability the child will be male who has the disorder. It doesn't tell us whether it's cystic fibrosis or polydactyl or what it is. It just says there's a disorder, a disease. Okay? And it just says determine whether there'll be a boy or girl and have the disease, and combine those probabilities. So how do we find out what is the probability of you having a boy or a male child? Anyone know? What's the probability of a male child? Yeah, that's right. So if I was doing this for 6 marks, I'd be like, right, I want to show I'm going to get 2 marks from this. Maybe 1 mark. But I'm going to get this right. So I'm going to be like X. I want to show the examiner what I know. So I'm going to. Now, if I leave it like this and just say 50%, I want to circle this so the examiner knows I know that is the boy, that is the male, and I'm going to give that 50%, right? Now I need to find out what's the probability of, brilliant. Now I need to find out 7 and 8. Let's check out 7 and 8. So if we look at the black square, the male has the disorder. They haven't even told us if it's recessive or dominant. This is how hard this question was when this one came out. It's a AQA question, by the way, for those of you that are wondering. The female does not have the disorder. The male has the disorder. How do I know if it's dominant or recessive? And they sometimes ask you to prove it from these diagrams. We'll check out these 2 parents here, 1 and 2. Let's check out 1 and 2. Look at parents 1 and 2. They don't have the disorder, but their kids do. Look, 1 and 2 don't have it, but number 6 does. What does that tell you? Recessive or dominant? Yes, it's got to be recessive. 2 dominant. If it's dominant, only a recessive can be passed on like this, right? So I now know 7 and 8. 7 must be heterozygous. That was the big problem. We didn't know whether 7 would be like, let's use big T, little t, or, oh no, they want big H, little h. So we didn't know whether 7 was big H, little h. We now know that 7, for those of you that are wondering, you will have scored 2 marks if you just work out that the boy is 50% chance. But now you need to. Now, by the way, don't give up if you're baffed, because they're going to do error carried forward on this. So 7 has to be big H, little h. Oh no, 7 ain't big H, little h, because we just said it's recessive. So what am I on about? I'm talking out my backside. We just said it's recessive, so the disorder. So we know 7 is little h, little h. We know 8 must be big H, little h. Why do we know? Now that we've decided it's recessive, because they've got a kid of each. So even if you mess this up, you're going to get the marks for correctly doing the Punnett square. So you could mess up whether it's dominant or recessive and still come out of this with 4 marks, because you're doing this. And now what we're doing, there's a 50% chance right? These have got the disease. We decided it was recessive because we had two parents without the disease who had a child with the disease. So they were carriers. They must have been carrying the recessive allele, if you were wondering. Number 1 and 2 we looked at to find out what it was, and we looked at their children. If you're given a pedigree diagram like this, always look at the parents somewhere and the children to see whether any parents who didn't have it but had a kid with the disorder, then you know it's recessive. Okay, what's the overall chance? 50 times 50%. Well, the overall chance of having a boy with the disorder is 0.5 times 0.5, and that is 0.25. Someone said 25% earlier. Yeah, so that's for the 6 marks there. You can write it as a percentage or you can write it as a decimal like that. Yep. Well done. Yeah. Nice one. Good luck. Yeah. It's a bit of a late one, and you might not want to stay up too late now because you might want to get good rest. If you've got geography tomorrow, you need to be getting to bed. If you just got the biology, obviously you might want to rest, get up and then work, do some exam questions. You might just want to flick through your revision guide. If you've got a revision guide, whether it's AQA combined, whether you're doing EdXL, whatever you're doing, whether you're doing triple AQA, you just take your revision guide. You might just want to test yourself. You might just want to walk around and test yourself. You can do any type of revision, really. You can go on to the past papers, look at past papers, but really you just want to make sure you've got knowledge of every single part, because it's about managing the exam. You can have really good knowledge on one, but you want to score points on everything. That's how. So even if you're not perfect or you don't fully understand something, you can score points on different parts. So, yeah. Couple more questions here. I should shrink that as I've got my camera right over it. So, how many chromosomes does a sperm cell contain? Human body cell has 46 chromosomes, but how many chromosomes in a sperm cell? Well done, Donald. Tasha, Cade, yeah, it's 23. Draw one line. Gene, which is the meaning of a gene? What's the meaning of a genome? I haven't talked about genomes today. And what is a nucleus? UC Gaming, I can tell you the exact percentage it will be, but well, it depends. What course? It depends which exam board you're doing. Genome is all the genetic material of an organism. Okay. So if you're doing AQA, you need to know why us understanding the genome is important. It's really important for education and learning about evolutionary relationships, finding out how similar we are to other organisms. It's also very important for medicine. If I know that on every single human, the cystic fibrosis gene is found on the exact same chromosome, chromosome 7, location 42, then I know exactly where to find it. If I know the eye color gene on every single human is found on chromosome 8, location BX5, then basically we've mapped the entire genome, all the genetic material of humans, and we know exactly where every gene is. We know where the gene is that makes your nose how long it is. We know where the gene is for how hairy your eyebrows are. We know the exact chromosome and the exact position on the DNA for that. And so it's very important for medicine and understanding genetic disorders, and it's important for education, research, and all these other things. The gene is, you should have gone with a small section of DNA that codes for a protein, and a nucleus is a structure which contains the chromosomes. Okay, this top one is a small ring of DNA. Anyone know what a small ring of DNA is? It's plasmid for that one. Right, over here we have fossils. This is called an ammonite. How did this fossil likely form? If you're doing AQA, you need to know this. So fossil in figure 1, how did that fossil form? Yeah, that's a good one. The ammonite shell was replaced by minerals. Why do we know that? Well, hard parts of organisms, bones, shells, teeth, get replaced by minerals. Okay, in mineral replacement. The traces is cast or impression. That's not a cast or impression. And the ammonite was frozen in ice. No, if it was frozen in ice, we'd still have its face, its eyes, and its little tentacles. So frozen in ice or stuck in amber or in a peat bog where it can't decompose, we'd still have all the other bits. Now, why is the fossil record not complete? The three ways, Narwhal, are mineral replacement, which is where you get hard sediment formed where the minerals used to be. You've got casts or impressions, where you get a print in clay or stone. And then the final best one is preservation, where decay has not happened. No microorganisms feed on the organism, and we have an exact image of it. We have its skin, we have its hair, we have its bones, we have its eyes, we know the color of it, we know all the information. These are very rare and very precious. Things like if they're trapped in amber, you have to not, decay mustn't happen, so it's got to be either low temperature where the enzymes can't work properly. It's either got to have no oxygen or no microorganisms or no moisture for decay not to happen. Yeah, you also need to know why we have no fossil record, guys. Why do we not have a full fossil record? And ammonite is one of these bad boys, Eggmaster. It's one of these creatures in figure 2. I've got a few here, but yeah, I don't want to get up and start picking it up. Okay, fossils. Yeah, some species have never been found. Earthquakes, volcanic activity, tectonic plate movements, geological activity. Well done, Gabriella. They have destroyed fossils, so we don't have a record. Of course, most organisms that have lived on the Earth do not have hard parts, so they've not been left behind. So no teeth, no bones. If you're an invertebrate, and most of the organisms on the Earth are invertebrates, like jellyfish, prawns, whatever, they've got shells, so they could leave those behind. Scrap that one. But lots of organisms don't have hard parts to their bodies, and they won't be left behind. Okay. Orsomenus. Yeah, already done. Looked at that. This is easy peasy, pudding and pie. What is estrogen? Yeah, so two statements which are true for sexual reproduction. Why don't I draw my line all over one of the questions? Went through a bit of the menstrual cycle earlier, but I'm happy to go again with it. Gametes are. I think there'll be a question coming up on it in a second. Oh yeah, that's no good, is it? If it's blurry, let's see if I can make this bigger. Hold on, one sec, peeps. One sec. Thanks for that, Gabriella. Didn't know if it was just my eyesight there. Okay. Still looks pretty blurry. Gametes. Yeah, 1, 5. Boom. Yeah, yep. They're not identical in sexual reproduction. They're not genetically identical, so not that one. Okay. Right, let me move this and see. Yeah, let's not bother with that one, but these ones with the hormones, let's see if you guys can do that. Estrogen, what does estrogen do? Sexual reproduction, so the offspring won't be identical during sexual reproduction. They're only identical in asexual reproduction because your children are made from two different gametes, two different parents, so different genes, the mixing of genes. Yeah, so estrogen, lining of uterus, whoever said that, but estrogen causes breasts to develop. That is a secondary sexual characteristic. Girls, estrogen is responsible for you getting hairy armpits, and testosterone is responsible for boys getting hairy armpits, and pubic hair on the genitals for both boys and girls, estrogen and testosterone. But testosterone makes boys' voice become deeper. So, secondary sexual characteristics are controlled by estrogen in girls and testosterone in boys. So, girls' breasts develop, their hips get wider to allow them to give birth. Boys, their shoulders here get wider when testosterone comes out, and their voice gets deeper, and both hormones create hair everywhere, which is a pain. Method of contraception. Cade, if you want to get rid of someone so you don't see their comments. Hi, by the way, this is a little trick in the chat. If you want to get rid of someone so you don't see their comments, tap on their name. If you tap on their name in the chat, it will come up remove or report. You could just remove, and then what will happen, you shouldn't be able to then see any of their comments then for the rest of the chat. So if someone's getting on your nerves, just take them out, man. Just go remove, boom. Yeah? And that's what you can do. You can just tap on them and click remove. And then they won't be spam. Then you won't have to see them anymore. Hey, can anybody do that? Yeah, I can block them. I can block them as well. But I'm just checking. Can you guys remove them so that you can't see their comments? Is that a thing that? Because I know I can do it. It says remove, report. Okay. Kay, did you manage to get rid of them? Yeah, so you can block them then. You can block them, yeah. Yeah, no, but I'm seeing if you can. Like you guys can click, and then I think you can hide user on the channel as well or something, so why is a stable biodiversity important? Why is a stable biodiversity important? Um, it's good for the ecosystem to have a wide biodiversity. It makes the ecosystem more stable, more likely to survive changes in the environment. So the secondary sex characteristics are genital hair, so hair on your private parts, hair on the armpits, for both boys and girls. For boys, its voice becomes deeper and the shoulders widen. And for girls, its breast develops and the hips widen. Um, Now, condoms. How do condoms prevent pregnancy? So, okay, hold on, hold on. I'm just going to remove NS, okay, because they're being a twit. All right. Let me just give me one sec. Yeah, they make a barrier, so they prevent sperm reaching the egg, and the oral contraceptive pill. Um, the oral contraceptive pill stops the eggs maturing. Give me one sec, peeps. I shall just remove someone. Um, One secondo. Oh my days. And let me give you guys, let's move on to this. All right, guys, see if you can see this while I just zing. All right, there you go. And I don't know whether I've actually removed NS or not. Has NS gone? Yeah. Okay, cool. Don't really know what I'm doing, as you can see, new to this. Right. Guys, adaptations. Yeah, no problems, no problems. Don't want people insulting people, like I wasn't sure whether you guys could just hide the person's comments. Like sometimes you can just click and hide their comments. Do you know what I mean? Um, yeah, so the wasp is structural, guys. Anything physical is structural. Claws, teeth. If you get a 6 marker on adaptations, structural adaptations are like claws, teeth, stripes, camouflage, fur color. All of that is structural, physical things on the outside of the body. Now, behavioral, uh, that's a behavior. They attract a female by banging on a stick. Hunting at night, that's all behavioral, so the way you behave. And then this is the tricky one, functional. If you need to discuss functional, they'll have to give you the information. Yeah, so functional is something that happens inside the body, like changing your metabolism, slowing down respiration for hibernation. That's a functional adaptation. If they give you a 6 marker, they'll have to give you the functional because you wouldn't be expected to know that, but you'd be expected to be able to spot structural ones like camouflage, thick fur, thick layer of fat to keep warm in the winter, small ears to reduce heat loss, things like that. And then meiosis, tick 2 boxes about meiosis. It would help if I didn't put my camera over it. Yeah, that's absolutely right. And Gabby, Arctic animals would lower their metabolic reactions in the winter in hibernation, and that would be a functional adaptation. So that would be pretty awesome if you came up with that. I don't know if they'd expect you to have to do that really, because I don't think they expect you to be able to do that. What do you reckon for meiosis? Cell divides twice. Yeah, I'll look at the decay practical. Tick 2 boxes. The cell divides twice. What else? Yeah, the new cells have half the number of chromosomes. Well done, yeah. Give the names of 2 other gametes. Sex cells, give the names of the 2 gametes. Easy peasy. Yeah, I can do pyramids of biomass. Egg and sperm. Awesome. And what's the process when egg and sperm join, guys? Oh, I've got no tea left. Deep. Right. What's it called when egg and sperm join? No, you don't need to know anther and stigmas. No. Fertilization. Nice. All right, give me a sec. Here is one on Carl Woosey and the old classification. You could get one asking you about. You could get a question asking you about. I've done classification earlier. Those of you that weren't on earlier, I went over Carl Woosey and Carl Linnaeus from the 1700s. You could get one with Charles Darwin thrown in there. If you're triple, You might have Mendel, the monks. You could have all four thrown in there, and they could say who came up with the theory of natural selection to explain evolution, and or they could say who came up with the domain system, or who came up with the classification system, and you have to know who came up with each. You don't need to know anything interesting, like, you don't need to know what their favorite meal was or anything, just what system they came up with. So, describe the three domain system. Yep. Jemima's got that. Archaea, bacteria, eukaryotic. So you might need to add a little bit of nuance to that and say archaea are ancient primitive bacteria found in caves or volcanoes or at the bottom of the sea, and it was noticed that they were different to modern bacteria. That's a bit of an explanation, really, rather than a describe. So you probably get away with just saying archaea, bacteria, and eukaryotic. Eukaryotic have a nucleus, and they are fungi, animals, plants, and protists. Right. What about the other question? What are the missing ones in the order? So, we've got kingdom, phylum, class, order, family, genus, and homo. Yep. No, sorry. Genus and species. Don't know why I said homo. That's a human genus, sapiens. Yeah. And then we just need to know what kingdom they came from. So, from the information. So the kingdom, they're a worm, so they're going to be in the animal kingdom, right? You have to work that out. And then what's their genus? What's their name? How would you know their genus? Gabriella is saying it is Spodoptera. How does Gabby know the genus? How's she worked out the genus? First name is genus, absolutely, nice one. Yeah. And notice the species is always in lowercase. Alright, awesome. Right, there's another one here. I will go over the decay. Let's just have a look at this one here first. Who devised this system of classification? The kingdom is animal. Basically, you just got to recognize what's an animal and a plant. I don't think they'll tell you if it's a fungi, but it will probably be animal, and you just got to know what an animal is. So yeah, insect, worm, and Linnaeus. Yeah. You got it. Woodsey came up with the domain system. Wallace and Darwin both came up with theories to solve evolution. Right, here we go, guys. Moose have distinct characteristics such as antlers. Went over this one kind of a bit earlier in the live. Describe how moose may have evolved to have large antlers. Yeah, animalia or plantae. Yeah, so you might want to say animalia as it's up there for X. That's the kingdom. Right. What's the theory of evolution? Natural selection, Dylan. Spot on. So you've got to explain natural selection. So your 5 marks for natural selection. Yeah, I'll talk about decay, and then we'll do the practical on decay as well. Give me a sec. Okay, Gabby's got the answer here. Genetic mutation. Outcompete the rest. As a result, you can pass on the genetic variant to offspring. Couple of extra things in there, Gabby. So we've got genetic mutations cause variation. Now, I would like to say this makes some moose have small antlers, some moose have big antlers. Okay? So we would say genetic mutation. That's right, Sana. But we need to then say means that we get variation. Some with small ones, some with big ones. We can take it from the question that the ones with big ones survived. We don't have to explain why, but you could try. You could say, oh yeah, they're better at beating up other ones or whatever, or they attract mates more. There could be any reason, thousands of reasons why the big antlers survived. But you just need to know that they survived. If they survive, you're more likely to have sex, reproduce, and have children, and pass those genes to the offspring, and then it's repeated many times. So yeah, mutation caused variation in the size of antlers. Absolutely right, Ali. And then that means that more likely to survive if you had large antlers, more likely to reproduce. 2 marks. More likely to pass your genes on to your offspring, and this is repeated many times. Yeah, that's right, Gabby. I don't think you have to say that, but explain it. But hey, cool. Right, this is another one on global warming. I'm just having a flick, seeing what other topics I haven't covered. I haven't done. Someone said biomass. I haven't done biomass. Trophy. What am I on about trophy? I haven't done trophic levels and pyramids of biomass or anything like that. But you can get ones there where you've got a scale and you've got to use the number of squares. But we'll go over that in a bit. Karam, you don't. Yeah, basically, it repeats over many generations, and eventually, all the moose have large antlers. Yeah, you could do. I did protein synthesis yesterday, I think, or the day before, but I can do it again. What would you do here, guys? Global warming causes sea levels to rise. Just any two other things that global warming does. Decreases biodiversity, changes distribution of a species. Boom, nice. Flooding, forest fires. You could literally list off 100 things. Causes extinction, destruction of habitats. In many areas of the world, household waste produces increasing. Give 2 reasons why we have more waste. Yeah, melting, ice sheets melting. Why do we have more waste? Increasing the standard of living, so we buy more crap. Okay, what else is causing more waste? Why every year do humans produce more waste? What's happening to the population? Nice. Yes, 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 yes. Well done, Jemima. Well done, Ali. Yeah, more demand for space. Perfect. Link to the population as well. Yeah, more cities, more houses. Right, storing carbon dioxide. Carbon dioxide can be stored in trees. Give one different way in which carbon dioxide is sequestered. Look at how they try to mess you up. That's a higher level question because they've tried to mess you up with language. Other than being stored in trees, how else is carbon dioxide stored, guys? They've used the word sequestered. I use a word for those examiners using a word like that. Nice. Sedimentary rocks stored in water. Awesome. Carbon dioxide dissolves in water. Perfect. And it does. It is in sedimentary rock. What's on the AQA syllabus other than trees? What's the other store of CO2? Other than deforestation, what's the other thing? Yeah, limestone. That's right. That's right. What's the other thing that other than deforestation, chopping? Yay, peat bogs. Someone got it. Yeah, so they're probably looking for peat bogs on the answer, but they will have to give you limestone. They will have to give you the shells of animals. They'd have to give you like prawns, lobsters. The shells have carbonates in them. They'd have to give you dissolves in the sea because the sea is a massive store of carbon dioxide. Yeah, so awesome. Couple more of these. I am getting to the decay. I'm just working my way through. I'm nearly at the end of these questions, so you never know. Right. Water cycle, dead easy peasy. Sana, what exam board do you do? Yeah, you need to know Peat Bogs, Sana. Peat Bogs, just while people are putting their answers down for 1 to 5 here, guys, I'll just go over peat bogs for Sana. Peat bogs are formed over hundreds of millions of years, so long, long time. And basically, trees fell down and were trapped and decayed in a way that they didn't actually decay. The plant material fell down and they formed this special area of land called a peat bog. There's quite a lot in Ireland, there's quite a lot all over the world. But peat bogs are very special habitats where lots of different butterflies, moths, dragonflies, lots of different organisms live, and they're perfect for those organisms. Peat is then used by us humans to burn because it releases heat slowly. But the problem when we burn it is it's got loads and loads of carbon dioxide in it because it was made from partially decayed trees and plants which absorb carbon dioxide. So it releases tons of carbon dioxide when we burn them as a fuel. So they're great to be used as fuels, but they release lots of carbon dioxide. We also put them on the ground because they contain loads of nutrients that make plants grow really fast and really well. And so we use them in B&Q and home base and any plant shops. We use them as fertilizer, and farmers put them on their fields to make their plants grow. When they do that, microorganisms feed on the peat and decompose them, releasing carbon dioxide. So basically, destroying peat bogs is increasing the carbon dioxide in the atmosphere and increasing global warming. But also it's destroying the habitats of those species that live there. Peat also can be worked into sort of the fossils kind of thing because peat is very acidic and denatures enzymes by having a very acid pH. And this means that bacteria and microorganisms cannot break down things that fall into peat bogs. So certain humans fell into a peat bog and got preserved when it was being, and certain animals have been preserved, and items. They found loads of things in peat. Anyway, let me just check. Everyone's on this. The only one that people probably don't know, which some of you do, is runoff, which is number 4. So number 4 is runoff, or if you're a geography person, you're probably asleep now. They call that percolation. Yeah, and Karam, if you're doing geography tomorrow, it's percolation or runoff, yeah, and evaporation. You guys have put up the answers. You guys are fantastic, awesome. Right, okay, the 6 marker is the Quadrats practical, but there's 2 Quadrats practicals. Again, I got another live where I did them. The 6 marker over here. This is Quadrats. Now, how do you know which Quadrat practical to do? You can use a transect, Gabriella. You would use a transect. Why would you know to use a transect? There's a keyword in the question that tells you not to do the random throwing the quadrat or the grid. Why do you have to do the tape measure or rope and a transect? What's the keyword, guys? There's a keyword. It's over a distance. I'm going to pronounce your name wrong. Yeah. Husman. I'll probably pronounce that completely wrong. Right. Yeah, you got that right there. So distribution. You see the word distribution, you're doing a line transect. So now you're going to just come out 6 marks though. Jeez, that's a bit deep. Students were asked to investigate the abundance. Ooh. It's a double bubble. They want the abundance as well. I didn't see that. I missed that. Yeah, so it's abundance as well, guys. So abundance and distribution. That's a bit of a double trouble. But yeah, that's cool. So really, if we're doing abundance or population, we normally do the random throw the quadrats about, use a calculator, make a grid, do a number generator, randomly place 10 quadrats, find the mean number of organisms in one quadrat, then divide the size of the field by the size of the quadrat and multiply by the mean number of organisms in the quadrat. And that's your 6 marks for population. Here we're doing a distribution, so we're going to do a transect. So we want to put our tape measure running from the stony beach to the sea. We want to place our quadrat at regular intervals. So I would say 1 every meter. You then need to really say identify the organisms in there, because what are we actually looking at? Distribution of different organisms. So identify the organisms using Google on your phone, using a key card, and then either use percentage cover. You're probably not going to use that. You're probably just going to count them up. So you're going to count the number of organisms in the quadrats, and then you can plot them on a graph. To be honest, we don't need to do the population one. They haven't mentioned population, so yeah. Yeah, population is mean times the total area. Yeah. It's regular in a transect, Ali Imran. You can say randomly select your starting point. So I could start, here I am. I could start there. Oh, I've kind of got my photo in the way. You could randomly select your starting point, so I could do it here. Like, whereabouts at the start of the stony shore do you want to do it? So you could randomly select the starting point, but you don't randomly place the quadrats. They're definitely placed with intention at regular intervals. You can place them one after another if you like. This is called an interrupted transect if you were doing A level. And it's called a belt transect when you place a quadrat on a rope. You can just put a rope down without any quadrats, but there's no point in talking about that one. It's one less marking point, so I don't like to do normal transects. I like to do a belt transect, which is where you place the quadrats. But at GCSE, you can just call this a transect. So place a rope, place a tape measure, 1 mark, across from the stony beach to the sea. Then place your quadrats at regular intervals, e.g. 1 meter or every 2 meters, more marks. Identify the organisms in the quadrat, more marks, using the word identify. Count the number of organisms in each quadrat, more marks. And then I would plot on a graph. We call it a kite diagram, but you can plot it on a graph. If it was the populations, if we were looking at population, right, Karam, you're looking for population. If you're going to do, if it said, so if it says distribution, if it uses the word distribution, you're basically going to do, if it uses the word distribution, you're going to do the transect. If it says estimate the population in the field, like if it says what's the population in the field, then you're going to do, let me just quickly go over that one, 50 meter times 20 meter. Right, there's my field. Here's my quadrat. So if it said find the population of buttercups in this field, then I want to say 6 marks. So we want to say randomly place our egg master. I'll come back to that, one sec. So you, if it says population, God, I'm losing brain power. If it says population in the question, then I want to do this one, and I'm going to say randomly place the quadrats. Now you can explain if you want how to randomize it. That's up to you depending on the marks. If it's 6 marks, or it asks me to, I'll explain how I'm going to randomize the quadrats. You could shut my eyes and throw them if you can't think of anything else. Or you can say divide the field into a grid, write coordinates, pull a number out of a hat, or use your scientific calculator to randomly generate a number and place the quadrat there. You want to place 10 quadrats. You then want to, i.e. buttercups, make sure you're not counting grass. You then want to say count up the number of buttercups, or estimate percentage cover. If there's too many, you're going to estimate percentage cover. Then you're going to say find the mean number, and my mean number is 3. So I found 3 buttercups in this quadrat. Yeah, pick a number from a bag or out of a hat. So I found 3 buttercups in my quadrat. How many are in the field? My quadrat is, you can say use a random number generator. Yeah, but you might want to say make a grid out the field. So my keywords are this. I want to talk about randomly place quad, count, identify is important. Count and identify. You don't want to get the wrong thing. You want to mention 10 times. You can do less if you want, but 10's a good number. You want to say, for more marks, find mean. And then the difficult bit, multiply by size of field. Yeah, so I go 20, hold on, 20 times 50. My field How big's my field? 20 times 50. My maths is awful. That's 1,000 meters, right? Whoops. How many quadrats fit in there? 2,000. So how do I know 2,000 fit in there? 1,000 divided by my 0.5 meter quadrat. So I've got 2,000 quadrats would go in that field. I know that on average there's 3 buttercups in the quadrat. So 2,000 times 3, 6,000. Okay, is the estimation of the population. Right. One other thing. Please, 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 please watch out for centimeters. All right. You need to times by 10,000. So if they do something effed up tomorrow and they say you've got a 25 cent, yeah, you've got a 25 centimeter square quadrat. If you have a 25 centimeter square quadrat, you need to turn that into meters. People think, oh, that's 0.25 of a meter. It's not. You need to divide it by 10,000 because there's 10,000 centimeters squared in 1 meter squared. So if you're given a centimeter square quadrat and you want to convert it into meters so you can use it with your calculation of the field area, you need to divide by 10,000 to convert into meters. So just remember that. That would be a bit too small, that quadrat. But yeah. The other thing on quadrats, before I move off of quadrats, quads, all right, guys. Okay. Watch out for the little squares. If you're counting grass or algae or lichen or moss or fungi, why can't we count them individually like buttercups? So this is a quadrat with little squares in it, okay? And that's supposed to be, let's call it lichen. You might have heard of lichen. Good, nice one, nice one. Too hard to count. Well done. Yeah, absolutely spot on. Well done. Too hard to count. Too many. How are you going to count something that's so small? How are you going to count grass? You're going to be there all day. Might be 1,000 blades of grass. So what we do is we do percentage cover. You must let the examiner know that you are taking account of half squares. So I would number this as 1, 2, I've got 3, 4, 5, 6, 7, 8, 9 squares. Let's call that 10. And then I might say this is a half and this is a half right on the diagram to let the examiner know. So I'm going to say I've got 11 squares covered. I need to work this out as a percentage now. What's my percentage cover of that quadrat? This is a 25 square quadrat, so I'm going to go 11 over 25. I'm going to times that by 100. Some of you will say, why don't you just do 11 times 4, as there's 4 25s? But you might be given something more difficult. So yeah. So it's a 44%, right? If I'm not mistaken. So 44% cover. The great thing if you're doing percentage cover, you haven't got to do the horrible calculation to multiply up. If I do 5 quadrats and I find out 44% is my mean, then I can apply that to the whole field and say, right, there's 44% lichen in that field. That's the population, 44% cover. So I don't have to multiply by the size of the field. Yeah, you need to know that for AQA. Yeah. Organisms in the river. The organisms in a river, that's a really good question actually. So let's imagine they try to screw you tomorrow and they don't use quadrats, and they say there's a lake or a pond, and they say you're looking at larvae, blood larvae inside, or nymph, whatever, inside the lake. How are you going to do that? Well, I'll need to calculate the volume of the lake, And then instead of using quadrats, I'd take cups, and I'd take like 5 cups, and I'd calculate the volumes of the cup, and then I'd find out the average for the volume of my cup. If I've done 5 or 10, I don't know, my cup might be 100 centimeter cubed, and if my lake is 100 meter cubed, then I've just got to convert my centimeters in. The stream might have died. Could all be over. It could all be over. I think it could be the end. I don't know if it's back. It's kind of dying. Um, um, I don't know where we got to. I've lost all brain power. Yeah. Do people want protein? Some people are saying protein synthesis. Do people want protein synthesis? Is that something people want? Do people want protein synthesis? I don't know how long this is going to stay alive for. Okay, protein synthesis. No, not in combined. I don't know how long this is gonna. The stream's gonna last. I think we've been on 3 hours. I don't know how long my probably my dodgy internet connection. So if I cut off, good luck everybody. If I cut off for tomorrow's exam, if it just dies on me, yeah. So, oh jeez, man, I think it's dying. I don't think it's gonna work. Server is having issues. Right. So transcription, going through quickly, guys. What happens in transcription is the DNA unzips. Why does the DNA unzip? Now, it does depend, because if you're doing AQA and if you're doing IGCSE, it all depends how much detail you guys need to go into. So if you're IGCSE, you got a lot more detail. Yeah, population of an ocean, you just want to find the overall volume of the ocean. And then penguin, you'd find the overall volume of the ocean. And then you would like have a bucket or some sort of container that you know the volume of. You would do 10 of those, find the average number of organisms, identify the number of organisms that are in each bucket. You know the volume of it, find the mean per bucket. Okay, now you do the same thing you do with a quadrat. You just take the volume of the ocean, divide it by the volume of the bucket, times it by your mean number of organisms found in one bucket, and there you go. However, the difference with volume is if you have centimeters and you're converting to meters, you need to divide by 1,000,000, whereas compared to area. Right, let me just see, because I've got a feeling with AQA, where is this? AQA. I'm not sure how much detail. I don't think you go into a lot of detail at all in protein synthesis. Do you do transcription and translation? You don't even do that in AQA. Oh, you don't even do transcription and translation. It's only in IGCSE they do transcription and translation. You do complementary base pairing, but you don't do protein synthesis. Now, yeah, you don't do transcription and translation, so you don't do that in AQA. Geez, they mention it a little bit. Okay, this is what they mentioned for AQA. So what you need to know is that a copy is made called mRNA. This is inside the nucleus, by the way. So this is the nucleus. If you're doing IGCSE, you need to know a little bit more detail about these steps. But if you're doing AQA, you don't. So a copy of the mRNA is made in the nucleus of the DNA. So my copy is this bit I've labeled mRNA, and it's just a copy of a small section that we call a gene. So it's a bit like if you imagine you've got a big textbook. If you've got your holy book or a really special book, you don't want it to, it's a precious, invaluable, super valuable book, you don't want it to leave your house ever and get damaged. You let someone come in and make a photocopy of one page and go out. So that's what it is with protein synthesis. A piece of mRNA is made, which is a copy of one section of a gene, just one gene. Now that gene then joins to a ribosome. Okay, now a ribosome, as you know, are found in the cytoplasm, and they're the site of protein synthesis. So what do you need to know in AQA is that carrier molecules, they have a special name, but you don't need to know that because you're AQA, so that's all groovy. Carrier molecules, which we call, which you don't need to know, so I'm just going to call it a carrier molecule. Carrier molecules bring different amino acids to ribosome depending on the code on mRNA. So depending on the code that's on mRNA, ribosomes will bring different amino acids. Carrier molecules will bring different amino acids. This is a carrier molecule bringing this rectangle brick, which is an amino acid, in. Now, depending on the code, you'll get different amino acids coming in to form the protein. Okay, it is called tRNA, transport RNA. That's the right name. It's called tRNA. It just says carrier molecules underlined in the AQA revision guide. So I don't know whether you need to know. It's called transfer RNA or tRNA. If you did want to know that, if you're doing IGCSE, you need to know that. This is tRNA, and this is your amino acid. Now, the tRNA has a triplet code known as the anticodon that binds to the codon found on the mRNA, and they're specific. So if it's CAG, the one above it will be GTC or whatever, but we don't want to go into that because, yeah, it's a bit too much detail. So, so that's in protein synthesis, which takes place on the ribosome. So what do you need to know? mRNA made in the nucleus is a copy of a gene of the DNA. It goes out into the cytoplasm, joins a ribosome where carrier molecules bring amino acids. The carrier molecules, if you want to know the name, are known as tRNA, transfer RNA, and they bring different amino acids depending on the code. Now, they actually join by complementary base pairing, which is the same pairing that the two polymer strands of DNA are joined together by complementary base pairing. So it's actually the same. You do need to know that in complementary base pairing, guys, what bases join together? Complementary base pairing, what bases do we have that join together? Anyone know? So you've got A and T, and you've got G and C go together. So A and T, G and C, and we call this complementary base pairing. Nice one, nice one, nice one. If I clear this, so what you could have is if I tell you that this is my code, A, T, G, C, A, G. Someone tell me what the other strand is going to read. So you might be asked, So write down what the other strand is. What's the other strand, guys? TAC, GTC. Nice. You guys are all over it. Awesome. No, not higher combined. This will be higher triple if you're AQA. This would be higher triple if you're AQA. I've been covering combined. There's loads of combined covered on this live. So, but this bit's just a bit of triple. So, you can rewind it. But it's like 3 hours long now. So, yeah, you can just. You guys are all over that. Nice one. So, but if it's mRNA, would it be U instead of T? It would be, but it's not on your specification to know that. No. Not for this one. Not for this one. For if it was the mRNA. But what I was saying is this is the two strands of DNA. So this is like DNA strands. So there wouldn't be U's in this. But if it was mRNA and I was making a copy to go out the nucleus and be read on the ribosome, both tRNA and mRNA have uracil wherever you were going to write a T. So what you do is you write it as a T and then you change your T's to U's. It can be quite confusing. You just change the T's to U's. Thymine becomes uracil on mRNA and tRNA, but you don't need that for your specification. Okay. You don't need that for your you don't need to know that. If you're doing AQA, you don't need to know that. But thymine is only replaced by uracil when you make mRNA. So for those of you that are interested in that, if I was going to copy this into mRNA and I was going to make an mRNA copy of this, every time you're going to write T, you write U. So I would go, oh, there's an A there. I would normally put a T here, but if it's mRNA we're making, I'm going to put a U. So I would go U, A, C, G. Now there's another A. I want to write T, so I'm going to change that to U. And then G, C. So that would be the mRNA. You don't need to know that. Okay, guys? The lives, yeah, it's been breaking apart. Yeah, Foxy, the mRNA moves out of the nucleus via the nuclear pores. So yeah, you don't really need to know this, but it moves via these little holes in the nuclear membrane known as a nuclear envelope called nuclear pores, and the mRNA moves out of those and then crosses the cytoplasm and attaches to a ribosome, but you won't need to know that. So M&A, antibiotic resistant bacteria, is the same as natural selection. So if you know your natural selection 4 points or 5 points, so mutations create variation, which means that some bacteria have a gene that makes them resistant. They are more likely to survive, more likely to reproduce by binary fission or asexual reproduction if you're combined, and pass on the genes to their offspring. And this is repeated over many generations until you have an antibiotic resistant strain. So, yeah. It's the same as really why do zebras have stripes, or why do sharks have fins, or why do some humans have blue eyes, because at some point blue eyes were selected for. So it meant that there was a random mutation that meant that some humans had blue eyes. They were more likely to survive, more likely to then reproduce, pass on their blue-eyed gene to their offspring. Why do some humans have brown eyes? Because in a certain environmental condition they were more likely to survive than the people with a mutation for blue or green eyes. So, and therefore survive and pass on. Any feature you see, why do I have 4 fingers? Why do humans have 4 fingers? Because blah, blah, blah. We do the whole natural selection spiel, the 5 marks. You do the same thing for antibiotic resistance. So, brief outline. M said, brief outline what we need to know about protein synthesis. is you just need to know it happens on the ribosome. You need to know that a piece of mRNA leaves the nucleus, combines with the ribosome, and then is tRNA molecules bring specific amino acids dependent on the code on the mRNA. So really, the code on the DNA gene that is originally copied to mRNA decides the protein that's made, because it decides the specific order of the amino acids. If you change one amino acid, it means your final protein. Remember, proteins can be antibodies, hormones, or enzymes. So antibodies, hormones, and enzymes all have specific shapes. If you change one amino acid, they may no longer work. So one mutation can cause those to no longer work. So it means the enzyme's active site may change shape and it will no longer bind with the substrate. It means the hormone may change shape, like insulin, and no longer bind to the liver and allow glucose to go in and be converted to glycogen. It means an antibody may no longer have the correct shape to fit the antigen on a pathogen and destroy it. So, yeah. No, you don't need to know. Punnett squares have been done earlier. You can zing back to the Punnett squares. We did them earlier. No, you don't need to know mRNA and TCAG if you're doing combined. Um, Brown eyes are dominant. They'll tell you that in the question. You don't need to know that off by heart or anything. Yeah, brown eyes are dominant. And just because something is a dominant allele doesn't mean that there's more of it. There does happen to be more brown eyes in the world. But some dominant disorders, you think, oh man, like loads of people have got to have them. If it's a disorder and it's dominant, it doesn't mean that it's more common if it's a disorder, because if the disorder is dangerous and it kills you, then it's going to be less common. So some dominant disorders or dominant mutations which lead to a disorder are actually really uncommon, because if you die very quickly, you don't get to reproduce. So no, I haven't. I didn't do no predictions for this. I did predictions for P1, which I got right. If you're doing AQA, I got the predictions right on my P1 live, but I literally didn't have time today to go through the B2 papers. What you got to do is go through the B2 papers. So to make a prediction, you've got enough time tomorrow. If you want to, you take out your CGP revision guide, you take it out, you go onto the AQA website if you want to make a prediction, because it takes about 45 minutes to make the predictions. So what all the people who do that all the people that make predictions, all what they are doing, whether it's Primrose Kitten or whoever, all what they are doing is taking the revision guide or the syllabus, easier to use the revision guide. They're opening up the exam paper and then go back to 2018, or you can even go back to the specimen paper 1 or specimen paper 2, and then just look through it, and every time you see, oh, structure of DNA, just write 2018 on your revision guide, where it is, and then 2019. Then you go through the 2020 paper and you write it down wherever, and then all you need to do is go back through and look for what hasn't been asked in the last 5 years, or what hasn't been asked in the last 2 or 3 years. Circle that and go, right, they haven't asked that, they've only asked that once in 5 years, or they've only asked that one, they haven't asked that ever. And then you go, ooh, that could be coming up. But they're only predictions. There are some things like electrolysis that have come up every single year, as you see in chemistry paper. Electrolysis has come up every single year. So do you predict it's going to come up again next year or not? But that's how they make their predictions. Well, no, if it's a recessive allele, Sneaky Trump, it's not going to die out over time, because recessive doesn't mean what people think it means. Recessive is actually means you can have a carrier, right? So I can have cystic fibrosis gene, but as it's recessive, it ain't going to kill me. That's great, isn't it? Because it means I can now go and have sexual intercourse with my partner and pass the cystic fibrosis gene on to my kid, so it's less likely to die out. If cystic fibrosis was dominant, then I would have it and maybe I'd die before reproducing and passing the gene on. So actually, dominant disorders are less frequent than recessive disorders. But dominant mutations that are not disorders, like brown eyes, are more frequent. Because when brown eyes meets blue eyes, normally the kids have brown eyes. But if that brown eyes was something that killed you, if it was a disorder, then all the people with brown eyes are dead right now. So it would be better to have the recessive disorder is more successful. Anyway, we don't need to go into that too much. I've done genetic engineering a little bit back. All you need to know for genetic engineering for AQA is, if you're doing AQA and IGCSE, it's two different answers. But if you're doing AQA, you just need to know that enzymes are used to cut a gene from the DNA of the organism that has the characteristic you want to transfer. You want to make, if you want to make, I don't know, a white bear. No, let's not do color of bears. But whatever. If you want to transfer a gene, if you want to make your carrots resistant to a disease that tomatoes don't get, you cut the gene from the tomatoes that protects them against carrot blight or tomato blight. You transfer it to your carrot. You use enzymes to cut the gene out, and then you cut the DNA of the carrot using enzymes, and you can then insert it using other enzymes, and you don't even need to know the names of the enzymes, and that organism will be a transformed organism and then have the characteristics. But you can say using a vector, where you transfer the gene to a plasmid first, and it can be a plasmid or a virus, but it's quite vague in AQA what you got to know. FSH and LH are produced in the pituitary gland, follicle stimulating hormone, and LH, not LSH. They're produced in the pituitary gland. Follicle stimulating hormone ripens the egg, and LH releases the egg. Someone was talking about the three things you need for decay. So the three things you need for decay: you need heat, you need moisture, you need oxygen. Well, there's really four things. You need microorganisms. If you don't have bacteria or fungi, nothing's going to rot or break down. They need oxygen for respiration. That's why you have holes in your compost bin. You need heat, so they need to get warm. Otherwise, the enzymes won't be at the optimum temperature to digest the material. I'm talking about the enzymes in the microorganism, so inside the cytoplasm of the bacteria or fungi. They're going to need heat so that they can be at their optimum temperature. And the other thing we need is moisture, because without water, the microorganisms will die as well. So your compost, you need to have all of that. Compost heap is just making a big pile of compost and letting it just rot down. Takes a long time. Yeah. Water is for hydration. So water is actually needed for all chemical reactions. So actually, when you talk about, you don't need to go into this level of detail, but when you talk about protease, the enzyme breaking down protein into amino acids, or starch being broken down by amylase into maltose, water's needed in every single one of those reactions. At A level, we call them hydrolysis reactions, hydrolysis. We're using water to break. So every digestion reaction that any living thing does, any time it wants to break something up, it uses water. That's why you'll die in 3 days without water, because it doesn't matter how much food you eat, you can't break the food down without water. So water's needed for digesting things, or just needed for chemical reactions. Plant hormones is not in AQA GCSE combined, like for phototropism. If you are doing triple and you're doing plant hormones, you need to know that auxins are made in the tip of the shoot and the tip of the root. And someone mentioned that. So let's just look at phototropism. Phototropism, if you're doing triple. Okay, phototropism, if you're doing triple, the light hits here, the auxins are made here, auxins are made in the top of the plant, the tip of the shoot or root, and auxins are growth hormones. What do auxins do when light hits them? Auxins move away from the sunlight, so what's going to happen is auxins are going to move to what we call the dark side of the plant, right? You can say they move away from light, and auxins' effect on shoots. Auxins cause cell elongation. You're going to get marks for saying that. You can just say cause the cells to grow. They cause the cells to elongate. Think about it. If this is the dark side and this is the light side, if this side grows more, it's going to cause it to bend towards the light. So these cells over here, they're going to get bigger. The auxins cause the cells to get bigger, and this is going to cause our shoot to bend towards the light like this. Why is that an advantage for the plant to bend towards light? How is that advantageous to the plant? I've already gone through the quadrats. No, it doesn't kill the auxins. Sunlight doesn't kill the auxins. They move away from the light, causing cell elongation on the dark side or shaded side of the plant. Yeah, more light means more photosynthesis, so it's an advantage for the plant, right? This is why plants are sensitive. They don't have a nervous system, but they use hormones to respond to their environment. So that is known as phototropism, responding to light. Positive phototropism, because the plant grows towards light. Here's the thing. This is a really key bit that people don't understand. Auxins cause cell elongation in shoot cells. They have a completely different effect in the root cells. So this is known as positive photo, can't spell, tropism. Positive phototropism. So once again, look out for the question. There's sometimes a clue. They'll tell you there's light in the question. AQA once asked a question where they said a plant was put in a box and then talk about how it grew, and people talked about phototropism. Of course, if it's in a box or in a cupboard, it can't get sunlight. So there's a clue there. We're not talking phototropism. Auxins are your hormone you need to talk about. They're made in the tip. They move down the plant by diffusion from high to low concentration. But if they get sunlight from only one side, they will move away from the light. And that will cause that side to grow more, causing the dark side to grow more and then bend the plant towards the light. We call it uneven distribution of hormones causes uneven growth, causing the plant to bend towards the light. That's phototropism. However, geotropism works like this, okay? So we can kind of do, I kind of could have done both at the same time, okay? So I can kind of go, all right. So if we're looking at geotropism, it's quite difficult because you can get geotropism in a shoot and a root. So if I take, let's take my plant pot. Okay, there's two types of geotropism. This is going to get a little bit messed up now. Okay. Right. Gravity. How do shoots grow up? I've knocked my plant on the side. This is for those of you thinking this is something weird. That is, I'm still dealing with the shoot, by the way, guys. I'm still dealing with the shoot here. Okay, so this is still the shoot. I'm not onto roots yet, but what's happening here is gravity now pulls down the auxins. They're made in the tip, but gravity pulls them down here in the shoot. And what happens here is my shoot cells. So this is geotropism of the shoot. Actually, let's put the shoot underground. Then we don't need light involved. In fact, let's clear this. Let's do this a little bit more sensibly. Right, because then I can do both just like this. This is the ground. Okay, let's just bust this out quickly. This is a seed. Okay. Let's deal with a shoot. You plant a seed underground. How does it know to grow up? Don't mention light. Light can't get through the ground. How does the seed know to go up? It's not because of light. It's not because of light. It's because gravity pulls the auxins to the lower side of the shoot. And then we get cell elongation on lower side. So we get uneven distribution of auxins, which leads to uneven growth of cells, which causes our shoot to our seed to do this and causes the shoot to bend upwards. This is known as negative, you can call it gravitropism if you prefer, or geotropism. So that is negative geotropism, and it's because we're dealing with the shoot. Pulls auxins to the lower side. I stupidly should have put of shoot. Now we get a big difference with the root. Let's zing over to the root now. Alright, now my root is going to be blue. This is a shoot. This is a shoot. Okay, now my root is going to be blue. Okay, so the root is blue for some stupid reason because I haven't got brown on here or like kind of root color. So then we're going to get a complete difference now. It's still geo. Gravity pulls auxins down. Exactly the same, but we have a different effect. What is the effect of auxins on root cells? Auxins inhibit growth of root cells. So you need to know the effect of the hormone auxins due to the different shape of the receptors on the cells. You don't need to know that, but the effect of auxins on root cells is different. They inhibit the growth. They stop the growth. What does that mean? That means the cells above grow more, like that. If the cells above grow more, they bend the root down, and the root ends up bending down. So the root's going to end up bending down, and we call that positive geotropism. Essentially, there's lots of similar points in both, because we can talk about geotropism. We can talk about whatchamacallit. You just really need to know that auxins elongate shoot cells and they inhibit root cells, and auxins move away from light but they move down with gravity, and you always end up with uneven growth and then the shoot or the root bending because you get uneven growth. So you get uneven distribution of auxins leads to uneven growth. I've done quadrats and belt transects before. Yeah, bending down is positive because it's growing towards geo. Remember geography, that's the earth. So it's growing towards the center of the earth. It's called positive geotropism or positive gravitropism, you're allowed to say on the mark scheme. Okay, I've done menstrual and classification earlier. You can rewind the stream for those. Yeah, inhibit means stopping it growing. Yeah, so you get different. Yeah. Another one, like if I took a plant now, just very quickly on this one. If you took a plant, I mean, this is never going to happen, but like if here's my plant, okay, with the leaves. What I've done is I've turned my plant upside down. Here's the roots. If you put a plant on its head, its roots are still going to grow down. And the reason being the auxins are going to pull the roots to the lower side of the auxins are going to pull the gravity. It's getting too late. It's going to pull the auxins to the lower side, inhibit the growth of the root on the lower side of the roots, meaning that the upper side is going to grow more and bend the roots down. And that is why if you put a plant upside down, its roots will still grow down, and that's positive geotropism. And it's because auxin inhibits growth in root cells but elongates and promotes growth in shoot cells, so it has a different effect. Biogenerators are let's have a little look. I think I'm dying now. Yeah, 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 yeah. Biogenerators. Biogenerators do decay. Um, Biogenerators, what do you need for a biogenerator? You just want some compost, some poo, some wheat, some waste. So you're going to put waste in there. You're going to need microorganisms. Uh, so for a biogenerator, you just put waste and microorganisms. We want the enzymes to be working well. So it depends whether you need any something to remove the heat. You don't want enzymes to denature. Let me just see how much you got to know on your biogenerators. Uh, oh, that's great. You don't really need to know a lot, so that's all right then. So your biogenerator, in different countries you could use this. They don't work in England, by the way. They don't work very well in England because we don't have the heat for the to create optimum temperature for enzymes for the microorganisms. So they just don't work that well. They're going to work better in hot countries. So in comes your poo and your wee and your waste. So put your waste in here. The microorganisms are going to do decay. We know that most microorganisms release carbon dioxide. That ain't helpful. But some microorganisms release methane. Methane is a gas, so we can burn that. So now what we can do, we can have the gases that are being released. Some are used for. So you could have gases like methane. And in some countries, they use this as a cooker, so the gas comes out and you can set a light and then you can cook your food or heat your house. Methane can be combusted for fuel, yeah, or for heat, or for cooking, fuel, heat, cooking, whatever. You can also take the waste sludge that's left at the bottom. The sludge can be used as fertilizers. So solid waste or sludge used as fertilizers. So you can then spread that on your crops and get your crops to grow. So yeah, so it's kind of useful, really. You can burn the gases that are released, and then you can use the solid waste to spread on your plants and make them grow faster. Methane, you can just call it in AQA biogas. It's obviously a hydrocarbon from chemistry that you burn in a combustion reaction. But yeah, that's a simple biogenerator. You do need to know the difference between batch and continuous biogenerators. This is triple, this bit. So your batch generator, you're just going to put a load of crap in there, seal it up, finish it, open it, take your sludge out, put it on there. That's more like, say, me doing it in my house if I wanted to create gas to cook with and stuff. And then I have to wash it out with a hose, get new microorganisms, new waste, put it in and seal it up again. And then this done in batches. Continuous is like if you're a big company and you're doing it. You're just going to say, let's have a pipe, and every day we're going to put waste in there, rubbish going in every single day, and every single day we're taking the gas off. It's a much more industrial sized process to do a continuous biogas generator. You need to supply it with waste every single day, and you also, if it goes wrong, you could be leaving it going wrong for like weeks, and it's contaminated and it's going wrong. At least with a batch, you only lose one batch. But yeah, batch is small scale, and continuous is on an industrial scale. Continuous generators are on all the time. You have to continuously supply them with waste, but they produce biogas at a steady rate. There is no real disadvantage. There are disadvantages and advantages, but batch generators, you just do them one lot at a time. It's kind of like, it's cheaper, easier batch generator, and if it goes wrong, you lose less. You lose less product, like you lose less stuff, because you're just doing it like one lot at a time. you do need to know that these biogas generators use anaerobic decay, so without oxygen, generally. And let's just have a look, guys. I might come on tomorrow, guys, to go through some other stuff, because I'm gonna have to get to bed. I am smashed. Pyramids of biomass or energy. No problem. Oh yeah, I can go transpiration in the phloem and that. What I was going to say is here, the you do need to know why pyramids are shaped like this. You need to know that at each trophic level, energy is lost. In fact, this is a very badly shaped pyramid. You would normally only get 10% passed on at each trophic level, unless it's in the sea where the producers aren't plants, they're algae. They have less tissues, no roots and stuff, so they're more digestible. They're single-celled organisms, and so plants tend to be able to have sea food chains or pyramids tend to have more levels because they're more energy efficient because of plankton or algae or whatever are more digestible. I can't even spell at this point. So guys, do you guys know why each trophic level or energy level of a period gets smaller? So you need to know that energy is lost at each stage. So you have energy loss through respiration. Yeah. Now the energy lost is through, if it's an animal, you could talk about movement. So herbivores, primary consumers are herbivores. They're going to be running away from the secondary consumers, which is the predators trying to eat them. And the secondary consumers are running away from tertiary consumers. So they're losing energy by movement. So they're also excretion. They're breathing out carbon dioxide. Carbon dioxide is made of atoms. It has mass, so they're losing that. That's not going on their body. They're also going for a poo, going for a wee, feces and urine, types of excretion, so that's not going on their body. If you eat a cheese sandwich, how much of the cheese sandwich becomes new atoms on your body? Not a lot, maximum 10%. You're going to walk to school, you're going to move about, you're losing that energy from the cheese sandwich. You're going to go to the toilet and poo some of it out. Some of it you couldn't digest, so you couldn't even access the energy because you don't have the enzymes to digest it, the fiber in the crust or the fiber in certain plant material. So excretion and movement. Respiration to produce heat. If you're a mammal like we are, feel under your armpits, you're making heat right now. So a lot of the cheese sandwich went into keeping your enzymes at 37 degrees, their optimum temperature. So heat production, movement and excretion. It's impossible for all the energy to get passed on. This is why there's so few lions and tigers and top predators in the world. You can't have a lot of top predators because you have to have a lot more energy in each level below. So yeah, which is why. And now the argument for being a vegetarian or a vegan can be explained using the pyramid of biomass or pyramid of energy, can be used explaining that. Basically, if only 10% of the corn a chicken eats, if you have 1,000 kilograms of corn and only 10%, 100 kilograms get passed on to the chickens in your farm, then you could make the argument, I've got 100 kilograms of chicken to keep some humans alive. But if the humans ate the corn instead of the chicken, there's 1,000 kilograms of food. I could feed 10 humans with the food I fed the chickens. So the idea is that only 10% is passed on. So with each step in the food chain, you're losing energy. If you're a meat eater, you're consuming energy higher up the food chain. If we didn't eat meat, we would have all of that. But I'm not a vegetarian, but I'm just saying that's the argument. All right, guys, going to sleep. Nice one. I might do a live tomorrow. So take care, guys. Good luck. Hit the like, share, subscribe. Bye.